Okay, you can if you want to, but otherwise we are going great means up
Okay,
till now.
okay. I think we'll be okay. Fine, no worries.
Yes, students are like participants are joining, so we can start exactly at 3:05. Is it fine?
Perfect. Perfect, perfect. I'll be
All
around
right,
and just let me know when we are ready. Absolutely.
all right.
It's login from two devices, I guess.
Yes, I have logged in from two devices.
Fine.
That's why I was worried about an echo, but I think there is no echo. Everything is
No,
okay,
no,
right?
no, there's no echo.
Great.
We'll just start in a minute, ma'am. There are participants who are joining.
Harry, please take care. You guys are better at organizing. I think you are the organizer. Just handle it yourself. Just tell me when you want me to begin.
Sure,
I'm
sure.
looking at my slides and fix it.
Okay, I think we can start, ma'am.
Okay.
Yeah. So, very good afternoon to everyone, the participants, Dr. Gitanjali Yadav, ma'am. So, today we are at day 5 of our course, and today's session is taken. I would like to welcome Dr. Gitanjali Yadav for today's session. I would like to introduce Dr. Yadav to the participants. Dr. Gitanjali Yadav is a senior scientist at NIPGR and the co-founder of Cementic Climate, a global citizen science movement for climatic action. She also serves as an adjunct professor of data sciences at ICER Bhopal and visiting lecturer at the University of Cambridge, UK. Dr. Yadav is an expert in genomics and machine intelligence with application in food security and conservation. She has a diverse educational background with a PhD in immunoinformatics from NII, a master's degree in biomedical research, and a graduate degree in botany from the University of Delhi, India. Dr. Yadav has received several awards, including the Hamid Fellowship from the University of Cambridge, Exceptional Talent Award from the Royal Society of London, DBT's IYBA Fellowship, INSA Medal, and the Shastra Obed Siddiq Life Science Award. As a co-chair of the International Data Policy Committee of CoData, Dr. Yadav strongly advocates fair principles and open access for data. She is the national coordinator of the DBT's iBreak plus Zero Waste Life program and promotes science in a way, as a way of life to students in schools and colleges. She has been a mentor for young scientists as one of the founder members of the Indian National Young Academy of Science and an ambassador for women in science at various national and international platforms. During the coronavirus pandemic, Dr. Yadav began conducting a series of free online training programs in data science under the name Protocols from Home in order to help students pick up vital skills in computational biology and make the best use of their time under global lockdown. This series is now delivered on weekends for bespoke cohorts of researchers. With this very short introduction, I would like to welcome Dr. Gitanjali Yadav to deliver her session. Welcome, ma'am, and over to you, ma'am.
Thank you, Tabassum, for that long introduction. But it is always a pleasure for me to come and connect with students. Particularly, I'm hoping today our audience is more students and less.
Almost students, ma'am. Only maybe one or two faculty members are there, and mostly UG students are there, ma'am.
Oh, wonderful. This is
Yeah.
great. It is particularly that you just mentioned that during COVID, I used to do these weekend sessions in, building capacity for students who are interested in doing computational biology. So, that was when I first began this putting together these e-materials, one of which you will see today, one of which we are
Okay.
going to be training
But
the
I must say, ma'am, this is a great effort, ma'am.
I don't do it so much now, but I would love very much to be able to restart some weekends. Actually, everybody has gotten so busy. So anyway, I'm going to try and share my screen. Please let me know if you
Sure, ma'am.
can.
Yeah, it's there, ma'am.
Right. Very good. So, good afternoon, everyone. This little tutorial session that we have today, as I was just mentioning, it is a result of a lot of interactions, a lot of teaching, and a lot of learning from various sources about how best we could address complex network analysis. And so I will be teaching this to you as students, and I hope as students, what I want more than anything else is a feedback on how easily you were able to understand what I'm trying to teach you, and whether you would be able to use it yourself in some analysis that you apply the tools yourself to some question that you're interested in. That would be really good. So before we begin, yes, of course, this is something very, very important. You might have seen in the brochure of today's event, and I really thank the Zakir Hussain College teachers, particularly Dr. Ruchi and right here Dr. Tabassum as well, for inviting me to be able to deliver it to undergrads. Usually in our research, we don't meet undergrads so much, and so it's a pleasure to be here with you today. You will have to install. It's not immediate, but we will have a little break today after an hour of lecture from my side, and then I would like you to I would like you to, at that time or even now, whenever you get extra time, please go to the Lord God Google and type in Cytoscape and download and install it on your device if you want to do the exercises hands on. Basically, this course is going to be in today's lecture and tomorrow's lecture. I'm going to talk to you about networks, and I'm also going to have small, small exercises in between, scattered in between, like practicals. Now, these practicals can be done on your own laptop yourself, just following what I'm telling you. And so therefore you could in principle do these things yourself. So that is why I'm saying it would be good to have the software. It's a free open source software. It has been built by scientists and students at the University of California, San Diego. And over the last 2 decades, a lot of people, uh, academics, students, scholars, have added packages to this platform. So therefore, it is completely free. The latest version is 3.10.3, but feel free to download whatever is compatible with your device. Okay, it takes a few minutes. Right now, as I checked this morning, it is Cytoscape 3.10.3. I used to have 3.9 on my device, so this morning I also updated my Cytoscape to be the latest version of Cytoscape. Okay, you won't need it till the tea break at all, so don't worry if you don't have it. We will just first understand the basics. The course is generally called Complex Network Analysis for Biologists. And here are some of the learning objectives. Okay, I've got to make my phone go on DND mode. Just give me a sec. How do you put on DND? Yeah, that's what it is. Okay. I hope you can still hear me. I mean, putting the phone on DND shouldn't completely cut me out of
You are
talking to
audible,
you.
ma'am. You are audible.
Brilliant. So, the learning objectives in this course are, first of all, whatever data you have or are thinking about, why should you visualize that data as a network? And if you do decide to visualize it as a network, how should you go about doing it? And before you go about visualizing your data as a network, certain basic concepts about networks, such as what kind of data sets are very amenable to network visualization, protein-protein interactions, DNA expression, transcriptomic gene expression data sets, as well as even sequences, biological sequences can be visualized instead of faster formats. You can visualize biological sequences as networks also. So a lot of interesting stuff can be done potentially with networks. It is, and I'm also going to teach you the basic. There are many software, but I'm going to teach you one, which is the Cytoscape, which I particularly like because of its being free and open source. And second, because, as I said already, it has been created by scholars. We will also look at certain layout algorithms and I'll share some tips that I've for effective visualization that I have myself learned over the last 20 years of dealing with networks. And finally, if time permits, I will also teach you a little bit about network clustering or partitioning, because it's very important to know what different things you can do once you've generated a network from your data. What kind of analysis can you do, right? So we will go there. But the most important bit is that the aim of this lecture and this little masterclass, I'll call it a masterclass in networks, the aim is not to finish the course. The aim is to understand whatever we are doing. That is more important. So let us not try to get to the next page of the slide. Let's just take it at our own pace. Feel free to ask questions if you don't understand something. And the Basum, my request to you will be, in case I have two screens, I can see the chat box here on the other screen. But in case I miss something, you can stop me anytime and say that there are some doubts that people have,
Surely.
and
Sure.
I will take care of those. There is also, just because, even if we do finish the current course or the tutorial or the lectures that I have planned for you today and tomorrow, don't assume that this is the end of everything. All I'm hoping to do is give you a primer into something new and something different in the beginning of a field. There is much, much more to network analysis. It's not covered in this course because this is considered as a beginners to intermediate course. There are many more things you can do with networks. For example, when you look at gene expression networks, which I'm hoping to make you generate and build and visualize for the first time over this course, the yeast interaction and yeast PPI and yeast gene expression networks is what we will see. But you could also then construct and analyze unweighted and weighted networks, and so those come under advanced, techniques such as WGCNA and so on. And then we could do consensus modules, eigengene networks, build vectors out of networks, and so on. And then you can compare networks, like you're comparing structures, or you're comparing sequences, or you're comparing any biomolecule of interest that you have. Can I assume that all the listeners today are life science students or from any of the natural sciences such as chemistry, physics, and basic earth sciences and other subjects, or do we have non natural science?
Ma'am, mostly they are life sciences, ma'am.
Very good, very good. So then that means you understand biomolecular data sets completely. You understand how proteins, DNA, RNA look like, how sugars look, how lipids look. And so therefore, you might think about how these interact with each other and how we might visualize them. There's also another level of network analysis, which is the bibliometry network analysis, which also we have a little course. I mean, we had put together some e-materials based on different places on the web. Everything is available on the web. And so therefore putting it together into a small e-material so that a lot of, so that students can understand it better is what we do. If this course is successful, we might think about doing more advanced courses. But let's see how you understand this one first, okay? So you need no prerequisites. Of course, you need to install the software. The aim is to make you understand this software and the concepts of network biology. You will need to install Cytoscape as well as certain packages such as the String app as well as the wiki pathways and so on, which we will do by and by. I will guide you step by step and handhold you through this installation. So please don't worry. In general, the session is going to be mostly lectures, like one-sided gyan from my side, whereas it will be 30 to 40% hands-on. So please do keep your devices on with you. Some of you may be listening to me on a phone, but I think it's better that you have a desktop or a laptop or some kind of or an iPad or some screen with you so that you can see, during the hands-on sessions, you might be able to gain a little bit more. But I've understood from the organizers that y'all are recording this session, and so therefore you can play it again and you can follow me. Maybe today just listen to me, and next time when you're listening, you could follow the steps step by step.
Uh,
And of
some
course,
of some of them, ma'am, they do side by side only because in last sessions they are doing like that.
oh, that's brilliant. That is
Yeah.
great. Actually, side by side is the best way because I'm right here for you to ask questions and sort out your doubts with. Yeah, so that's wonderful. And the last thing that I want you to know is that we are going to work at our own pace. So there's going to be no rush. There's going to be no pushing to finish this part today or that part tomorrow. We will do whatever is most comfortably handleable. So, of course, complex natural systems permeate many aspects of everyday life, medical science, social science, but more importantly, life sciences, biology, chemistry. And in the last couple of years, or at least in the last 10 years, much of research papers, if you've read the research papers that we see, a lot of real world system structural properties and dynamic properties are now visualized as networks. In general, you think about genes or proteins in a network, but there is so much more that can be visualized as a network. This tutorial that I'm going to be taking with you, it will introduce you to the basic concepts of network theory and some of the most widely used global platforms. Our aim is mainly to conceptualize, to make your conceptualization better for your own data, whether it is textual data, genomic data, protein data, and so on. Just wrap your head around being able to understand topology and how can my data be made topological. Okay, it's like taking biology to geography. And so, therefore, some of the basic stuff that we will cover will be the fundamental concepts of biological networks, the software, which is Cytoscape, data visualization, and network partitioning. The main goals and motivations that we have for this course, if you do it completely today and tomorrow, you should be able to identify relevant types and sources of these networks. You should also be able to identify what kind of approaches you need to take to visualize your data as a network better. And you should also be able to look at a data set and say, I can convert this to a network. I can look at this table, take this table out, and instead of presenting this data as a table, I can present it as a network. And you must learn how to publish, share, and export your networks out to the wider world. Okay, so we are doing this course over 2 days. In general, we do it, in shorter, like for the PhD program at NIPGR, I take 1 hour over a couple of weeks. So it goes slowly. But with you, we are going to do it today and tomorrow over a little bit of an extended session. So we will take a break of 15 minutes at any time that you start to get tired. Okay, but generally after an hour, not before that. And so therefore, the first bit right now is setting up the you know introducing the concept to you, setting up an installation of the software and so on. And then, of course, we will be creating and visualizing networks today. Right. So this is again the first bit. I will, of course, give some homework and DIY, and it's up to you whether you want to do the homework and DIY. The good thing about it, or now that you have come and you are attending this entire session of classes and lectures and hands-on sessions that you're getting an introduction to, I would say make the most of it. And do the DIY and do the homework. And because I will be here tomorrow also, you're more than welcome to ask me questions here. All right. So first of all, introductions and ice break. A lot about me and the course. I would like to know about you. I did put into the chat box a little message saying I'd like to know what you're interested in. So please do put an answer to these two questions into the chat box. What kind of species? If you close your eyes and somebody said, "All right, you have the freedom to do whatever you want in research, natural sciences, life sciences. What species would you like to work with?" And the second question I'd like to know is if any of you have already used this software before, because if you have, then I know how to, what kind of speed to be taking with you guys. Okay. So I'm going to give it a couple of few seconds maybe to look at the chat box while seeing your responses. Wait, I can't see the chat box at all. That's interesting. Okay, I think I can. Oh, very nice. There are lots of answers. All right. Umama is doing BSc at Zakir Hussain. Yeah, all that is great. Where are the species that you might be interested in? Do I have any species that anybody is mentioning? Okay, Kriti Bodke likes E. coli, but has not worked with this software before. Very good. All right. So lots of answers. All right. When we have a break, I'm going to look at a little more of these. And there's somebody who would like to work with Triticum and Tista has never used the software. Right. That is good, Tista. In that case, we'll be looking at, wow, tardigrades. Very interesting. So when I was in college and somebody asked me this question, I mean, from my school time only, I had wanted to work with orchids. And I thought if I ever become a scientist and if I ever do research, I will spend my whole life working with orchids. Of course, I've now spent more than 25 years as a researcher at the different leading institutions in the country, in the world. I never returned to that childhood promise of mine. I mean, I still love orchids and I still would like to work with orchids, but God, there is no time. So I've been working with a lot of species. But it's good that I see very interesting species named by some of the students on the Oinothera lamarckiana, Chavi. Very interesting. I'm glad to see your interest in, from most students, interest in very interesting species. Very nice. Keep putting your questions or comments in the chat box. Any the way, carrying on. The motivation is that most biological processes in cells, in us, outside of us, in the environment around us are never performed by a single independent macromolecule, right? And so it is always a collective group team effort. And the fact that this is a collective group and team effort also means that therefore, unless you look at the whole picture together, you don't really get the picture at all. So you've got to come out of the box and you've got to be able to see things as they are. So therefore, looking at a system from outside the box is more important, and that is where complex networks come into the picture. In the next 10, not 50, 10, or even less than 10 years, 20 years, if biology has to be summed up into one word, it would be integration. We have to integrate all sorts of information, not just genes, not just proteins, not just different kinds of biomolecules, but much, much, much more. And many of you already are probably spending more time on the computer and the internet to generate and describe your data or understand your experiments or to analyze what other people have done and find relevant data for comparison or existing knowledge in your field and relate it to the stuff you're interested in. And if at all you come to publishing some of your work someday, you would still need to be doing more integration with existing information. So therefore, it's very, very important that you understand that there are different diverse data sets in biology. For
Uh,
example, as you can see on the slide before me right now, you
Gitanjali,
have
I'm
your
sorry.
data, could be tabular data, textual data, spatial data, genomic data, proteins information, microarrays, transcriptomes, RNA-seq, and so on. Nowadays, we also have bioontologies, we have reaction chemistries, mechanisms of reactions, pathways as independent nodes in networks. And of course, literature-based knowledge extraction is another level of looking at data, and we are looking at fair data much more than that we ever did in the before the last 10 years. And so, therefore, on the other side of this slide, you also see my favorite little species. I've been still looking at a lot of you putting species. There's somebody, Kishan Gupta, is interested in Arabidopsis thaliana. Arabidopsis तो पौधा भी नहीं है. So what is it that makes you interested in Arabidopsis, Kishan? I would be very interested in knowing that. And of course, there's the dire wolf that somebody is interested in. So I'm interested in clammy. Clammy is Chlamydomonas reinhardtii, this unicellular alga that you see on the right side of this slide. And the reason I'm interested in clammy is that despite this guy being just a single cell, there is so much data that is already publicly available for clammy, such as the nuclear and organellar genomes of clammy, such as the synchronized diurnal transcriptomes of clammy. Diurnal would mean around its entire diurnal life cycle. Every half an hour, gene expression has been calculated, measured for every single gene, and it is recorded and publicly available, published in different records, not once, not twice, but multiple times. You also have the nuclear and organellar time resolved transcriptomes. You have the stromal proteome, you have the chloroplast proteome, you have the ribosomal proteome, you have the interactome, but not just the interactome, which means not just the interaction between proteins, but the spatial interactome. So the geography of the interaction is also known for clammy. And of course, the unique thing about clammy is this little microorganelle which doesn't have a membrane. It's called starch plates all around it. It's called the pyrenoid. Those of you who are botany students would know about the pyrenoid. The entire proteome of the pyrenoid is completely delineated for clammy, the flagellar proteome, the centriolar proteome. That means the subcellular microorganellar proteomes and transcriptomes are already available, as well as the phosphoproteome, which means the phosphorylated proteins. Their subset is also completely measured and publicly available for researchers to look at. So, if a single cellular organism can have so many data sets, think about your species. Go back to your answer that you put in the chat box and think about your species. It's a eukaryote, it's a multicellular eukaryote in most of your cases, and therefore there would be so much more data potentially that you could extract for it, right? And therefore it makes sense that it's you should not be looking at one kind of data at one time, but to be able to superimpose these data sets. And for that you need network analysis. Biological network analysis originated, from the tools and concepts that we had in graph theory and statistics and social network analysis. Graph theory, of course, was a set of abstract concepts and methods that could be used to visualize and analyze biological networks long, long, long ago. In 1735, Leonard Euler came up with this little bridges puzzle, and that set the basis of the graph theory altogether about the whole idea of topology. And so after 1736, when Euler came up with the idea, for 200 years, people weren't even looking at it very seriously. And then in the mid 1930s, social network, social studies, and social scientists came into rediscovered Euler's work and said, "Oh wow, we can, rather than going on with long paragraphs and subjective kind of detailing of topics, we could in one go visualize all our data as a network." And then in the 1940s, it was psychologists and then anthropologists and then other computational people came in. And in the 1980s, when the IBM PC first was invented and came into the market and people started using network codes, then basic network topologies with the LAN and WAN topologies started to be built on the graph theory. And then, of course, people going on, and even till now, biologists have not woken up, they're still sleeping. Then at the turn of the millennium, it was physicists that came into who discovered that, looking at data in the form of a network would be great. And even at the turn of the millennium, biologists are nowhere. So it's just now in the last 10 years that we have now begun to see that every single paper that is published, every single course that is being taught in leading universities around the world, there is a lot of focus on networks, right? And so therefore, it's very, very important that you too are part of this idea of grasping or wrapping your head around what is a network. A network is nothing but some nodes and some edges. Okay, now the nodes could be a set of actors, which could be you and me, individuals or people, but they could also be biomolecules in life sciences, such as genes and proteins. The nodes are also a set of collections. So each node could be one individual or one gene or one protein, but you could also assign nodes as collections. So each node in a network could be a whole species, or could represent countries, or could represent departments in your university and my university, as well as entire genomes. Okay. And now these nodes, which you think about as circles or units in a network, have to be connected by edges. And so there are sets of ties or relationships that connect the nodes. The nodes are individuals, people, genes, actors. These actors are connected by ties or relationships, and the relationship is what defines that entire network. The relationship could be a positive relationship, and the relationship could also be a negative relationship. Okay, can somebody think of a positive relationship as compared to a negative relationship in terms of biomolecules? If yes, put it in the chat box. Each kind of a relation, as I said just now, defines the network that you're looking at. So if the network is about people and about you and me knowing each other, then it is an acquaintance network, right? If all of you are using Facebook, you might be having some friends. I don't know, these days youngsters don't use FB. You're all on Insta, aren't you? So on Insta, who are your friends, who are not? So you have a network of friends on Insta, and that becomes your social network. Okay. But if the network is about two actors, which are people, not that they are friends, but the fact that if a line is drawn between you and me, then we both are co-authors on a paper. Then this network is not a social network. Then it's not an acquaintance or a friendship network. If our relationship is that of having co-authored a common paper, then that network is a co-authorship network or a bibliography network. Okay. In the same way, if all the actors are genes or proteins, and if they are proteins, and if they are like if proteins are interacting with each other, then it's a protein interaction network, or a regulatory network, or a co-expression network, and so on. Okay. And in the same sense, a negative example which I had asked earlier is could be gene expression. Now you're looking at five genes maybe, and two of those genes are helping each other through a positive feedback loop, whereas three of the others are inhibitors in some sense. So that inhibition is considered as a negative kind of relationship. Okay. In the same way, yes, Chavi, brilliant. Symbiotic relationship could be positive. I mean, symbiotic are generally positive, but there are also negative relationships in the ecosystems around us, and predatory relationships, parasitic relationships. So those as well are relationships, and they can be represented. Commensalism, brilliant. Kavyanjali, lovely name. And so these things are absolutely there. And so these relationships can be represented on a network, and you can have your own network based on your own data or whatever interests you. Okay. Now, why should be looking at your data as a network? Why not happily look at it in a textual or a tabular format? First of all, looking at your data as a network gives you a very powerful tool to reduce the complexity in your data. Also, it is 100 times more efficient than looking at a table, because tables get boring after a while and everything starts to fly over your head or besides your, around your sides, and it doesn't get absorbed inside. But looking at a network, looking at some colors, looking at how things might be, um, as a network is much more efficient. It is also a great way of integrating data and visualizing data in an intuitive way. Why intuitive visualization is important? Because then you can, especially for biology, you can immediately come up with, when you look at a network and you see, oh, this is how the relationships are, which you wouldn't have recognized through a table, you can immediately say, oh, I could do this with it. I could come up with something else. I could ask this next question. So in research, what is most important? It's not to find an answer to a question. I think every question should lead to more questions, which your teachers, I'm sure, have been telling you. So the whole cycle of research is not a self-closing cycle, but tangential paths into newer and newer places where you could go to by looking at data in the form of a network. All right. And networks, of course, are all around us. You have molecular networks. You might have cell to cell communication happening in all multicellular organisms. You have your nervous systems made up of axons, all of them connected to each other as a network. And of course, there's the social network between us, right? Now, why should you care about network visualization? You should care about network visualization, or particularly learning how to convert your data into a network. It's because it's never just the elements of a system that matter, but also how those elements are stitched up together and put together, right? Integrating diverse data sets meaningfully is another very important reason why you must try to address your data as a network or visualize it as a network. Networks can quantify these relationships that you have. If it's a co-authorship network, for example, where you have two people being authors on the same paper, there could be another two people who are authors on not just one but five papers, and therefore the quantity or the weight of the relationship is also very important. Now, the three of us are friends, but you and I are far better friends than, me and somebody else outside of this system. And so, therefore, the quantification of these relationships, which is enabled through network visualization, is also a very important feature of understanding the system better. You can gain insights at the species level, at the population level, as well as at the systems level by addressing, by considering or visualizing your data as a network. You can find new patterns, you can create new hypotheses, which is more important. As I said earlier, the whole idea of doing research is to reformulate your problem in a newer way, in a better way, and to be able to readdress or re-ask the same questions better and better. Has anybody studied prompt engineering in AI? I mean, this is a Hatke question completely, but prompt engineering is a mechanism by which you could get better answers. So you could get a machine to give you in the human machine interface or the human machine interaction. You can improve by learning how to interact with the machine, and therefore reformulating the problem is really, really important. And if none of you have done the prompt engineering, there are lots of free courses available online. You should do it. I know that students use a lot of chat gpt and ecosia and OpenAI and DeepSeek. But if you've not learned prompt engineering, you've not really achieved the full power of what AI stands to offer you today. Okay. Coming back to networks in biology, particularly, certain biological systems, all biological systems, but certain others can be represented far better using the theory or using the tools that graph theory gives us. For example, there are food webs that you must have studied in kindergarten. You must have studied about species interaction networks when you were looking at ecology in your undergrad courses, maybe even now during your masters, you're looking at these neurological networks, metabolic networks, signaling networks, cell to cell interaction, genetic interaction, gene regulatory pathways, as well as protein protein interaction. All of these and many more, many more relationships in biology can be represented as networks, as you can see on this slide. Okay. In cell biology, particularly, here is a little example of a network. Here, what you see now, let me explain this to you. So here's a network. It's just a random rough kind of network that you. Okay, I'm gonna hide this. How happened? Okay. So here in this network, there are certain nodes and there are certain edges that are connecting these nodes. Now I'm going to ask a few questions and I will keep an eye on the chat box at the side so I can see what you guys are responding to me with. Okay. So here are nodes, and there are two kinds of nodes in this network. The nodes could be proteins, but the nodes could also be the products or metabolites. All right. And then there are certain kinds of relationships between these nodes. So um, and each kind of relationship or each kind of edge between the nodes is colored in a different color. So if you see blue colored edges or blue colored lines, then that means that the nodes that are being connected by those blue colored lines are so if the blue colored lines are protein protein interactions, and therefore the nodes that are being connected by these blue colored lines have to be by default, of course, proteins, right? Because proteins only can have protein protein interaction. So here, who is going to tell me how many proteins are there in this network? You see a small network. It's just a rough kind of random sort of example network. And the proteins are all represented as circles. And Preeti says there are 4 proteins, but Krati says there are 6. Palak says there are 6. Manisha says there are 4. All right, I need one answer. 6 and 4 will not do. Okay, write down the names of these proteins. Do you know the names of these proteins? The answer, of course, Arshia, you are not right. Shreya is right. Oh, Arshia is not right, but Shreya is right. And Preeti has answered it brilliantly. It is A, B, C, D, E, F, my dear. Absolutely. Shivanshi, brilliant. So there are 6 proteins, but some of you said only 4. Do you know why? Because in your mind, you're only looking at 4 circles that have a lot of interaction going on between them. So at the first instance, any new person would say 4, but I'm very glad that some of you could at least answer the correct answer, saying it is 6 proteins. Absolutely brilliant. Now, the next question to you is about this simple network. There are 6 proteins here. Are all the relationships between proteins only, or is there any kind of particular relationship that is also between a protein and a non protein moiety? For example, a relationship or an edge that exists between a protein and a metabolite. What is that? And can you give me an example of it from this network? Because let me tell you now, the AB relationship is a protein protein interaction because it's blue in color, and I told you that already. Now, the B-D relationship, for example, between B and D, you have another protein protein. But there are certain relationships, and Kavyanjali has got it already. Amarjeet also understands it perfectly. There are also relationships that you are seeing here, which are between metabolites and proteins. Now, think carefully about, yes, you're absolutely right, Anushka, Palak, brilliant, correct answer. Now, for those of you who have not understood what I'm trying to get at, what I'm saying is that this data that you see in just one picture in front of you could have been put in a tabular format, and you would have a protein protein interaction table where you would have 6 or 7 rows, more than 6 or 7 rows. A interacts with B interacts with C interacts with D, but B also interacts with D, and D also interacts with E, and it goes on monotonously like that. In the same way, there would be another table saying that A is basically a catalyst protein, and it's taking an input of M1 and creating a product called M2. And M2 becomes a substrate to the protein B, which is making another product called M3. Now, that would be a metabolism data, which would be in the metabolic data file. And then you would have the protein protein interaction data file. And then you would have a regulatory gene regulation data file. Then you would have a cell signaling data file. So, looking at 4 or 5 different files is one way. Of course, that is a way of looking. And traditionally, we have been looking at multiple tables. But if you took all of those tables and put them into one single picture, then this single picture tells you all of that together. And this is why looking at different data sets, which becomes monotonous in a textual or a tabular format, would be great to, it completely transforms the way if you add some color, add some, some design and a kind of topology to that network. So here, for example, if I ask you how many kinds of relationships, how many kinds of different data are you looking at in this picture in this network? So one kind of, let me tell you that one kind of data set that you're looking at is the identity of a biomolecule. It could be a protein or a metabolite, but you're also looking at metabolic data, like who is the substrate and who is the product. You're also looking at gene regulatory data. You're also looking at protein protein interaction data. Can you tell me what else? How many different kinds of edges are we looking at? How many different kinds of relationships are there in this network alone that you are seeing in one go? Yes, Kavyanjali, brilliant. Amarjeet says 5 types. What are the 5 types of relationship, Amarjeet? Chavi says for Bhumi, Arshia, Anushka. Anushka also thinks there are 5 relationships. I want to know what is the fifth relationship because I think there are 4 relationships, but I may be wrong. I would like to know what are these 5 that some of you have seen. Others are agreeing with, others I do agree with that there are 4 types of relationships here being metabolism, gene regulation, cell signaling, and protein protein interaction. If there is a fifth type that I have missed, please list it out here and I will modify my slide and I will make sure that next time when I meet students like you, I would know that there is a fifth type. Okay? Moving on, so now some of you have an idea about how a network looks like, what kind of information we can put, and how different kinds of information can be put into the same network. All right? The other concept that you need to understand is that of network partites. Now, we here, those of you who are ecology students or who have ever studied ecology or wildlife or biodiversity would know that there are certain bipartite networks in in the in general in nature. For example, plant pollinator networks. Plant pollinator networks are bipartite networks. That means there are 2 kinds of people or 2 kinds of nodes that can interact with each other but not with themselves. So, for example, if you think about a plant pollinator network, the pollinator pollinates the plant. The pollinator doesn't pollinate itself because pollination is something you do to the plant, right? And a unipartite network is somewhere where potentially, theoretically, every single node can be interacting with each other. So unipartite network, give me an example of a unipartite network, a protein-protein interaction network. Potentially, if there are 5 proteins, theoretically all 5 can interact with each other. Whereas if you have 2 bees and 2 flowers, then it cannot be that the 2 flowers are pollinating each other or the 2 bees are pollinating each other. It's basically bee to the flower and flower to the bee, right? So you don't have a self interaction. So then you have these multipartites. And unipartite would be when you have a single, when everybody can interact with everybody theoretically. Now, I want you to type in the chat box an example of a unipartite network. Can you think of something that is unipartite other than a protein protein interaction network? Please tell me. Here there are examples of k-partite networks in AI. When we are doing a lot of these days, we are building a lot of transformers and we are using neural networks within these. So there are lots and lots of neural network graphs that we use in artificial intelligence, and there you have multiple partites. So different layers of the neural network come after one after the other. So in graph theory, which is a kind of mathematical subset, a k-partite graph is a graph whose vertices are or can be partitioned into k different kinds of independent sets. Independent sets means they can be interacting with each other there. So, for example, if you have a co-authorship network, would a co-authorship network be a unipartite network or a bipartite network or a multipartite network? What do you guys think? Put it in the chat box and we'll have a discussion about it. Okay. Moving on, there are other kinds of networks in cell biology, which could represent co-expression, sequence similarity, co-annotation, disease association, drug-ligand interactions, and so on. Also, there are applications of biomolecular networks that you could think about. Now, what would you do with a network? Now you have a gene function prediction network. You have a gene network, and you want to predict function. When you predict function, you can identify guilt by association. Okay, and when you are detecting protein complexes, you can use subnetworks or network clustering that tells you which complexes are probably more, which we will study tomorrow, because tomorrow uh, we are planning to do protein complex protein network partitioning. Okay, and I'm very happy to see Krati's answer. Yes, indeed, it's a unipartite network when you look at co-authorship networks. Krati, brilliant. Network evolution is something you could study in a biomolecular network. When you do biomolecular networks, for example, when my students are building networks, they build networks over time. So, so if you're looking at the cell cycle of a cell, and you build a network in the first few stages of the developmental stages of an organism, and then you move on to midway, and then to the vegetative, to the floral transition, when the reproductive stage of the organism sets in, and then when at the end of life, when there is the later developmental stages. So at each of these stages through time, you would have a different network, or you would have the network evolving in a different way. So we've done network evolution for Chlamydomonas reinhardtii. We've done network evolution for a couple of other organisms that we've been interested in, mostly crops, but also microbes. Okay. Tista has an interesting comment here that needs me to stop and read it out. So, in the World Trade Networks, where countries can be nodes and the trade routes are the edges, or maybe the interbank network, where banks are the nodes and the loans between. Yes, brilliant, Tista. This is very good. In fact, you remind me of an intern student who had joined me like 7 or 8 years ago. And this kid comes in for just a couple of months to work in the lab. And we were interested in how certain invasive species have become have invaded different parts of the world. So we have current today's data on where this invasive ageratum connozoides is present in different parts of the world. What the student did was she picked up world trade data, and she said, we don't know when this species came here, and we don't have historical records of the species because it's an invasive. You wouldn't have specifically somebody bringing it and making a record of introducing it to here. Of course, the British in the British times, there was so much interest in the colonial days. There was so much interest in gardening and plants and moving plants across the world that there are a lot of records available. Very neatly built records are available if you wanted to. But when did a species come into which new place? You sometimes can find between these locations trade records. And so she picked up the world trade records and she overlaid it on the Google Maps of locations where the species has been occurring, has been found to occur, which information you can find in the GBIF, which is the Global Biodiversity Information Facility. All of this data is freely available, but scattered in different places. So this student brought it up together, put it together, and we could visualize how ageratum moved around the world from the 1800s, from when we have the world trade records, over the 1900s and so on. It was beautiful. Yeah. So good. Very good, Tista. Absolutely. So these data sets can be represented as networks. Then, of course, you can do diagnosis for diseases based on subnetworks. You can understand pathway flux movements as well. More often than not, when I find students listening to my lecture, one of the major questions they have, okay, I have a question, and maybe you've taught me how to get the answer, but where is the data? Am I to sit and generate the data first? Or is there a place that I can go to and get the data and hit the ground rolling? Like I've learned network analysis today, and I want to come up with a question tomorrow, and I want to start working on it day after tomorrow. Can I get that? My answer is yes, of course you can get it. You can get your data from among more than 500 different databases that are publicly available for you to search from. Now I'm going to give you a few examples of some of these data sets, particularly in protein-protein interactions. So protein-protein interactions, because you're all life scientists and biology students, you all are very well aware that protein-protein interactions are physical or selective contacts that happen or take place between pairs of proteins in certain molecular regions, of course, not everywhere, and sometimes, more often than not, in a defined biological context. Okay? And so, what is an interactome? The interactome, like the genome or the transcriptome or the phenome, is the totality of protein-protein interactions that can take place in a cell or in an organism or in a specific biological context. So when I say we're looking at the spatial transcriptome, spatial interactome of the pyrenoid, means that you have a specific biological context. You're looking at the pyrenoid inflammate, and you have a spatial context. You're looking at only this microorganelle. And of course, you have a very specific organism context because it is clammy. And then it happens inside the starch bound micro location called the pyrenoid. Okay, so the PPI network, therefore, is a graphical representation of a group of protein protein interactions in which proteins are represented as nodes, and whether or not they physically and selectively contact each other is represented as an edge. Now, proteins can be interacting with each other transiently or long term, right? So they could be, we call them mathematically as date hubs and something else. Okay, so long term or short term interactions. And you can see some of these examples shown on this slide below, but we will see some by building ourselves today when we look at Cytoscape. There are different detection methods for protein-protein interactions. There are high throughput methods and low throughput methods. The Y2H and the APMS are some of the common methods that we have. Also, X-ray diffraction studies, and then yeah, but no single method can accurately reproduce a true binary interaction observed under physiological conditions, because every interaction detected experimentally is basically fundamentally artifactual. And so, therefore, you need to have multiple evidences. So, if you're looking at two interactors and looking at a binary physical interaction, then you should if you're looking at, if you've got two interactors, you should look for a binary interaction. But if you have co-localization, that would just come from proximity of multiple people in the same room. So all of us being in the same room studying this course today is a co-localization kind of PPI. Then there could be n-ary associations where multiple proteins are in a single complex. Okay, and then of course you, such as the electron transport chain complex and so on. Then of course you have functional or direct interactions such as enzymatic interactions. And so these are binary interactions are shown to you on the slide, one-on-one interactions. Co-localization is when everybody gets together and these proteins come together to do something interesting. Then you have n-ary associations where there are more people making a complex or something specific for that particular day, such as you see on the right side of this picture with the SAM complex and a TOM complex and so on. And then, of course, you need to know how you would represent protein-protein interactions, which have individual interactions, have domains, interacting domains, as you can see on the slide here. And so these domains can be overlaid on the sequence itself. Where do the reactions happen? Where do the interaction happen? And once you overlay these ranges on the sequence, you can build interesting kinds of networks out of them. And all of these databases that are currently available in the public domain called interaction databases are mainly of two kinds. One are the primary databases where where papers which are published with something new go into one place. And then there are two other kinds of databases, such as the predictive databases and the metadatabases. So I don't know how many of you are aware of all these primary. What kind of interaction databases are you aware of at all? Are you aware of the UniProt? Are you aware of the PDB? The primary databases, as you can see on the slide, and the metadatabases that you can see on this slide are so the most common primary one is intact, of course. It has molecular interaction data that has been manually curated to very high detail. And when I last looked at it, there were more than 800,000 protein interactions there. And these were coming from more than 14,000 different papers. And each result in intact will be revealed as one line of evidence coming from one paper. And if there is more evidence for the same interaction, you will have multiple lines of evidence or multiple rows in your data. And all of the data can be freely downloaded from Intact through the website or through FTP or through Scikit, which I'm going to teach you today. Okay, and then you can curate this platform. It's been done very neatly already, and sometimes there are double curations also for quality control and so on. So another example of a primary database which I used to work on during my PhD was BioGrid. And then you have the predictive database. I don't know how many of you have used string, but a lot of PhD students and research scholars these days like to use string because they feel it's very user friendly. It's got a very nice GUI interface. And of course, it has like millions, almost 10 million proteins from 2000 different organisms that can be found there. And there are so many different kinds of evidences that you have for two proteins or two genes to be interacting, such as experimental, gene fusion, co occurrence, co expression, and so on. So you should try out the string website, but if you haven't done, don't worry, we are going to do it together today. This is the string visuals, and you also have the metadata. The metadata sets, metadatabases are databases that take information from multiple primary databases. Because you see, if I'm your supervisor or if you're doing research on your own, also, you come up with an example. So you say, all right, I'm interested in, let's say I'm interested in leukemia, and I want to study all the proteins that have been known to be involved in leukemia. So, of course, it's a human cancer, and you're aware it's connected to the blood cells, and so therefore you would need to have knowledge of, either you would have to read up all the papers that have been published in this area, and of course you cannot do that because you might not have access to half the things, or even if you do have access, you may not have the time to read all the papers. Even if you had the time to read all the papers, you may not have the knowledge to understand each paper brilliantly. And so therefore metadatabases already look at these different primary databases and primary papers and put together aggregated information coming from different sources. And so therefore, you can be sure when you're using a metadatabase that you're not missing out on information. I'm interested in working on leukemia. I want to find what I can find from it, but I first need to know what else is already known. And so I don't want to miss anything just because I need to look there and because I don't know what databases are available out there, right? I'm an undergrad student. I don't know much. I've only today done this course on biological networks. And so therefore, using metadatabases allows you to overcome the limitation of not having enough knowledge about different databases. And so these different metadatabases are therefore, such as, for example, IREF index is very, very good. And again, results are shown as pairs of proteins or pairs of genes which are interacting with each other. And there's always an evidence and provenance of that data, whereabouts that you get when was it published that these two interact with each other and so on. Also, then comes Psychic. Psychic, I really appreciate, and a lot of students I find don't know about Psychic. It's the unified query client where all of the primary as well as the metadatabases are taken into account to be able to address the query of whether A and B interact with each other, or for my particular species of interest, such as the what was it, the orchid, or for my particular disease of interest, such as, let's say, leukemia, I can find out all the information that is currently known out there in the world, so that what I do or the research that I do is not really reinventing the wheel, right? So one of the metadatabases that uses this unified query client of Psychic is called Mentha. It is literally updated every single week, and it holds protein interactions aggregated from multiple primary databases. There are more than a million protein interactions that they have. There's no specific focus on protein or any species, but quite a few species are there. Other kinds of, so what kind of choices would you have if you're interested in PPI and if you want to do a small project, let us say in your college or in your school or in a research institution? You want to work with non-model organisms. Let's say you come up with, okay, I don't want to work with a Arabidopsis, I'll work with a species that is a real organism. And if it's a non-model organism, you're okay with fuzzier kind of data, but you need the highest possible interactome coverage without wanting to go for just the yesterday's paper or day before yesterday's general idea. So then you should look at string. But if you are interested in model organisms, because the maximum kind of data is available on model organisms, there is a bias in the published world that most of us as researchers prefer to work with the organisms that are model. So there you can find experimental data and you would have the highest possible coverage, and you wouldn't want all details, but you would want everything to be covered. That is where you go for Mentha. And then, of course, you can go for intact when you want, when you're open to either model or non-model organisms. Okay. And these two, the Mentha, intact, and Mimex are entirely covered within the Protamic Standard Initiative Common Query Interface, which we otherwise know as Psychic. Okay, so you will learn how to use Psychic today. Here is an example of a protein protein interaction network with undirected edges. 80% or more than 80% of the proteins that are there in this network are connected into one giant cluster called the PPI network. Do you see some specific clusters within this network? Can somebody look at this network carefully and tell me if you see any clusters or subclusters which might be protein complexes or which might be some kind of a sub network? Maybe it is located to a particular area, just the nucleus or something. And some of you have said yes, you do see these networks. How many sub networks or how many clusters do you see in this network? This is what we call this is a general real world network effect, where because everybody is not connected to everybody but a lot of people are connected to a lot of people, you have the small world effect. The small world effect means that every node is connected to every other node through a, has anybody seen that movie called 6 Degrees of Separation? So the movie said basically what was said in that movie, and it's not the name of the movie, it's a fact known before the internet explosion. We all knew 6 degrees of separation basically means that every human being on this planet knows every other human being on this planet through a step of six people. So you would know every other individual on a planet through six people, which means you know somebody who knows somebody who knows somebody who knows somebody who knows somebody else, and that distance of six people covers every human being, regardless of national borders, borders of barriers of language, barriers of culture, barriers of religion, barriers of whatever. So, and that 6 degrees of separation was a fact before the internet. So, after the internet, which has brought so many of us together, I believe that we now have an even smaller world, which means that maybe today you might be connected to every other human being through a step of to a number that is smaller than six. Well, I can't say that for sure, but I'm sure it is. So, any the way, the idea of a small world network is that some people will be connected to a lot of others. So, there'll be hubs, right? Even in your college, you might have noticed some people are particularly, the center of all things happening, okay? And certain other people like to stay aloof and on their own, and they're not particularly they're, I won't call them introverts, but they like to be solitary, okay? So what happens in a general network, in a real world network, is that 90% of the nodes are such solitary nodes, and a few are hubs. That means connected to more and more others. And these hubs tend to expand and they tend to bring in more and more solitary nodes to connect to themselves. And then these hubs become important parts or important features of a network. And eventually they will evolve into the most important interaction within. So if it's a college network, let us say, or if it's a national network, even if it's a trade network, you would have certain hubs that certain people who are nodes who are far more important than certain others because they are communicating with far more other nodes than most nodes. Okay, so that's called the small world effect. We will come to it when we are looking at the graph theory concepts today, a little bit later. But I need you to look at this network and understand that in one look, when you see that most proteins, first of all, there's an overall interconnection. Everybody's connected to everybody somehow through somebody else. And then some are connected with fewer people, whereas others are hubs. So this is a small world effect. Okay, here is an example of here are some properties of protein-protein interaction networks with a small world effect. Small world effect means um, you would have interconnected binding, which could indiscriminately spread perturbations. And how can individual pathways therefore be turned on and off without upsetting the whole system? That is how you'll get to understand the small world effect. So here is a network with about 20 nodes, and each node is like a circle, and there are some connections between them. I have colored I mean, I have colored two of these or labeled two of these nodes as A and B, and there is a there is a connection between A and B. That means A is connected to B through how many degrees of separation? Who's going to tell me how you can reach B from A? You could take a step to the, I don't know if you can see my mouse. From A, you go to the next blue guy, that's step one, then another step two, and another step three, and a fourth step four, and a fifth step five to get to B. So you would take 5 steps from A to reach B. So A and B don't know each other directly, but they know each other through 4 other people on the way. Okay? And so this, hey, what happened? Right? So that's because there is a small world effect here. Also, this network is very non random, of course, but it has in general 3 distinct clusters. Will you agree with me or will you disagree with me? Do you think there are roughly 3 clusters to this network? Yes or no? And I'm looking at the chat box. Chavi says yes, Kriti says yes, Arshia, Palak, and Tabasum say yes. So yes, indeed, there is 3. So how does this knowledge of these 3 clusters add to your understanding of the network or your interpretation of the network? What I would say from a network communication point of view is that it is very, very important for network robustness or a network's health or a network's survival. I would not use the word survival, for a network's robustness to be able to communicate with all the nodes. So there should be absolute available communication between all the nodes, but it need not be direct communication. So A and B are not directly communicating, but if there is a message to be passed through the network, the message can go from A to B in 5 steps through 4 people who will be the mediators of the message from A to B. And if something goes wrong, for example, this is a biological network and there is some kind of a pathogen or a targeted attack on the node A, and the rest of the system has to protect itself, then it would basically cut off this edge between the A cluster or the A subcluster and the rest of the 2 subclusters. There is a single bridge edge that you will see between the 2 nodes that are there. If you, I don't know if you can see, let me see if I can get a highlighter or a laser pointer. Yeah, maybe now you can see the laser pointer on the screen that I have created in my PowerPoint. So between this node and this node, I would call these bridge nodes. I would call this a bridge on the network where all the communication from one cluster, which I will call the subcluster A, goes into cluster B or subcluster B and this third cluster, niche wala. And if you want to break the communication, then this single bridge is all you need to break so that if something gets infection, there's an infection in the A side and you don't need to bring it to B side, you can break this. The system can protect itself by blocking this interaction. This understanding I need you to have for when you look at a network, you should immediately see which are the most important nodes, which are the most important edges, and which are the bridges that will mediate flow of communication between the nodes. Okay, so let's take an example, a quick primer into network analysis to understand whether you guys understand network topologies and network interaction. So when you look at a network, the way the nodes are placed in relation to each other node is called topology of that network. Okay, so if any of you have been to India's leading immunology institute called the National Institute of Immunology, then the 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 building, if you look at it from a drone or from the aeroplane upstairs from the sky, you would see that it looks like an antibody. If you come here to the National Institute of Plant Genome Research, where I live and work, and if you look at the institute from the sky, you would see that the institute looks like a set of petals. And the topology, therefore, is is the is the physical shape that you visualize in the first instance. And so if all the nodes are connected to each other in a daisy chain, and the last node comes back to hold hands with the first node, then we call it a ring topology. If all the nodes are connected in a daisy chain, but the last node is not connected to the first node, then we call it a line topology. If the nodes are connected to each other like a dendrogram in a phylogenetic structure, then we call it a tree topology. If the nodes are sitting on each side of the main backbone, then we call it a bus topology, as you can see on this screen. If all the nodes are connected to one central hub or one central main guy, then we call it a star topology. Sometimes all the nodes are connected to everybody else. That means there is nobody who's not connected to nobody. I mean, there's nobody who's not connected to anybody. That means everybody's connected to everybody. Okay. So that is called a fully connected topology. Okay. So here we are today in the classroom, in this virtual classroom. I want you to close your eyes and think about our class. You don't know online who else is there. Well, you do know. You can look at the participants list. But suppose we're sitting in a room, we all don't know each other, but all of you are listening to me. So all of you do know me. What is the topology of our network, our classroom network? Out of all these topologies, which of these topologies would you say is our topology? And I'm looking at the chat box. Oh my goodness, brilliant. Oh my goodness, brilliant. So most of you have said have said that our topology is the star topology, because of course you're putting me at the center of the star and all of you are connected to me. But I find it very interesting that Tabassum has answered. So the thing is, all of you probably know each other also. So in truth, maybe we are the fully connected network, and so it would be correct. But quite often, because we are on a virtual class, we don't know who else might be there. We might not know each other so well, so we would be on a star. So it is correct. Both of these answers are true. We could be the star topology. We could also be the fully connected topology, depending on whether you know each other or don't. Okay? Give me an example of a line topology. Put it in the chat box if you can come up with it. Either way, going on now, this is a random network. And on this network, each node is shown as a circle, and each node has a label or a number or a character letter that is the name of that node. And of course, there are certain lines or relationships that are connected to Krati. A family is not a line topology. A line topology would mean that one person knows the second, knows the second knows the third, but the third does not know the first. So yes, Nayan, I agree. That's true. Cascade is one. And the other is what I had said about the 6 degrees of separation. You know somebody who knows somebody else, who knows somebody else, who knows somebody else. And that way you wouldn't know the third person, but you are connected to the third person through the middle guy. And that is a kind of a line. Yes, exactly, Kavyanjali. So coming back to this network, I want you all to look at this network. There are about 10 or 12 nodes. There is also about 10 or 12 edges between these nodes. Now, let us say this was the network of what is the example I usually take in my classroom? Yeah, let's say this is the example of a pathogen, of a very serious pathogen of the of Homo sapiens. You are the researcher or the scientist who has to crush or kill the network of the human pathogen. Which node, and you don't have, nobody has all the infrastructure to kill every single gene or protein on the pathogenic network. You have a chance, you have the opportunity to only kill one of these, mutate, knock out, or do something to one of the genes. Which gene would you delete, and which gene would you knock out to be able to do maximum damage to this network? Is my first question. So I can see on the slide that most of you have automatically, your brains have wrapped around the idea of hubs and isolated nodes, and all of you are saying that it would be the D. Bhumi has a very interesting answer where she says the CD edge is more important than just D alone, and I appreciate that very much. And then Nayan has something even more interesting, which I find really. I would love for you to type out why you think the J node is most important, Nayan. Great. So, okay, most of you are saying D. Let's take D as the main one. Now, let us say this was the network of the Indira Gandhi T3 terminal, right? And you are the security officer, the head of security of the Indira Gandhi T3 terminal, and all of these different nodes are the gates of the different flights or whatever else, different departments in your airport. This is the T3 airport, and you're the head of security. Where on which node would you put maximum security, because you are mapping out threats. Your job is to map out threat, and your job is to protect the network, and your job is to know which one to protect. Therefore, where would you put maximum security, the largest number of guards, the maximum number of, yes, it would be D. And the reason you would be putting it as at D is because you will assume that if the attack comes, it will come on D only. But exactly for that reason, guys, the fact that you as an attacker, terrorist on the either you could be a terrorist who's trying to harm the T3 airport, or as the head of security who's trying to protect the network against potential terrorist attacks or potential threats, you would put the maximum security on you. And therefore, even if you're the pathogen, you're not the head of security, but you're the, let's say, COVID pathogen, or you're the HIV pathogen, you would put maximum protection or security on the node D. And the fact that now you know that D is therefore the most protected one, then would you still want to attack D? If you have limited, if you are the guy who has to, you're the scientist who has to work on this, and let's say it's the HIV network, and you've got to come up with a fail-safe method to be, damaging HIV in the best way possible, and you know that D is most protected, then targeting D does not make sense, because you will put so much energy, you will put a knockout, but knockout karte hi D might be having backups, multi backups, third-party backups, and so much more. So therefore, it makes sense. Therefore, network analysis is for you to know that instead of the most visibly important center or hub node, maybe I can kill something else that is also going to distort or damage this network, but it will not take so much of my energy, because I have limited experimental or effective bullets or tries, and I want to damage. So when you say you want to damage a network, or distort a network, then what you want to basically say is you want to fragment it, you want to cut the communication there. And if the communication here happens through bridges, as you just saw on the previous slide, then you've got to break the communication. And for you to break the communication, therefore, removing D is a great idea, but breaking the bridge between C and D, or between D and J, or removing J, as Nayan had said, would completely destroy. Removing J would be easiest because it is least protected, because it doesn't have many connections. Getting rid of J would immediately cut the network into two different pieces that don't interact with each other. And then you could take out PKNL on the other side, and then you could come and start to look at how I might damage C and then make it worse. So what I'm trying to say is, when you look at a network, when you look at data in a visualizable form, you're in a much better position to make decisions or predictive decisions about what nodes, what subclusters, what edges, or what bridges to break or to make or to build or to create um, um, uh, additional help on. So here again, if you randomly target this network, randomly what will happen, because there are not enough hubs. If you do any random node failure on this, on random attack on this network, you will end up hurting it very less. You will not make a great damage. You'll probably randomly hit somebody called A. And that still maintains the overall communication between the nodes. That means the 6 degrees of separation, or the 4 degrees of separation, or the 3 degrees of separation in this case is still intact. You could randomly hit another node, you would still not touch the communication, you would still not disrupt this communication in the network. You could randomly remove this third guy also. So imagine back to back random hits are happening, and yet the network is robust against random node failure. And that is the property of a real world network. But of course, when you have targeted node failure, as all of you said it initially, you would target node D, and it would completely immediately disrupt communication. What would targeting node D do? In theory, if you could really get rid of D, you are absolutely breaking or fragmenting your network into at least 3 pieces and maybe 6 pieces, because the F, E, and M would be isolated individual nodes falling off the radar altogether. So D is truly the best way to disrupt the network, but D would be most protected, and they would need much more of your energy and much more of your infrastructure and much more of your time to disrupt. And so therefore, I would suggest that the edges are better. Okay, now what we are going to do is we are going to talk about the software Cytoscape, but I think that I have been speaking for a long time. If there are questions, you can ask me now, but what we might do is we might take a 10-minute break or a 15-minute break. Tabassum, would you like to suggest what kind of break we might take?
Usually we take
Or
15
should we
minutes
wait a few
break,
minutes and do the
ma'am.
break at 4:30? What is the suggestion?
Yeah, we take a break of 15
I
minutes
think what we will
usually.
do is we will carry on for a few more minutes so that I can tell you the background of Cytoscape and then leave you all to download and install Cytoscape.
Maybe
How about
maybe
that?
you can tell
Let's do
us
that.
how to
Huh?
download it, and meanwhile in the break some of us can download
So
it.
I'm going to talk about Cytoscape for the next exactly 6 minutes, and then we are going to take a
Sure,
little comfort break where you can grab a cup of tea or a cup of coffee or some juices I have here with me.
sure,
And
sure.
so there are many, many, many visualization tools. Can someone hear me, or have I been kicked out of the group? I think I'm still there. Okay. There's Bahek, there's Cell Illustrator, there's Bayana, Giphy. So all of these visualization tools, some are licensed, most of them are free. And it depends upon what kind of visualization you want to do. And of course, if any of you has used R for coding, there are lots of packages in R that do brilliant network visualization. But we will be looking at Cytoscape because it is cross-platform, it is open source, it's a huge consortium of academicians, and it lets you do so much, it lets you play around so nicely with so many different kinds of things. Okay. So it's free for both commercial use and academic use. It is developed and maintained by universities and research institutions. Most importantly, the University of California, San Diego. It is also allowing you to use tables and networks interchangeably. Everything gets saved as Cytoscape files, which can be formatted and converted into different between different platforms. There are hundreds of plugins for analysis of specific kinds of data, such as proteins or genes or species or genomes, and so on. You can also export the network in various formats. You can export graphics in so many different ways, even as vectors, and that's good for publications. Okay. So the best thing about Cytoscape is that it first of all allows you to do network topology analysis, which is the first thing you should always do. When you look at a network, you should look at its topology. What kind of topology is it? Is it a ring? Is it a mesh? Is it a star? And therefore, what do I know about it? How do I understand it? Wrap your head around it. And these topology statistics usually are measured by certain centralities or mathematical quantified values, which will be facts about that network. And these could be node degree, degree distribution, centrality, and so on. The other thing you could do with the network in Cytoscape is you can address the modularity, which refers to the identification of subnetworks. Remember when you were looking at the picture that I showed you for a PPI, and many of you could immediately detect subnetworks or subclusters in that network? So that is done there. So you could represent through modularity in Cytoscape, you could represent your molecules physically or functionally coordinated to achieve a specific function within the bigger network. The third thing that Cytoscape allows you to do is motif analysis. Now, motif analysis is something that all of you might have heard about or read about in sequence analysis. But motif analysis is like identifying patterns that are overrepresented when you are looking at a network. And when you look at the randomized version of the same network, you would not find those patterns. So only in the real scenario would you find those patterns, and therefore those motifs might have a biological meaning or some kind of value or some kind of importance. And it is good for you to find that motif. It is utterly near impossible to find that motif if you look at tabular or textual visualization rather than network visualization. So regulatory genes and regulatory elements are often composed of such motifs, which make it very interesting to study them. The last thing you can do with Cytoscape, oh no, not the last, but among the many things you can do with Cytoscape is network alignment and comparison. This is where you can use tools and packages that identify similar things between multiple networks. Also, it can help you to study temporal, space time relationships, evolutionary relationships, especially between protein networks of different organisms. The same thing. For example, if I'm interested in photosynthesis and clammy, and I want to look at the the flowering or floral transition network in different species, Then, based on what information I have for each of these networks, I can compare the floral transition network of an orchid with a legume, with a wheat grain, with a grass species, and so. Okay. And there is a huge community, so that if you need help, you don't have to come running back to me or to your teachers. You can just go to the Cytoscape community and they'll be very happy to address your questions and answer your queries. There are three basic steps that are required for data visualization with Cytoscape, and that are integration, analysis, and visualization. Okay, so you integrate data which comes from various places, you do the analysis, that means you find the various centralities that we will study today, and finally you do a visualization of your network. So this is what you're going to be doing now in the next session, which I hope will be the hands-on. So this is it. We have reached the absolute center point of today's talk. And so we are going to begin. I want all of you to set up and install Cytoscape on your devices. You can do it by going to the Cytoscape download. This is the link that is there. This is December 2021, but I think this is absolutely June 2025. So this is June 2025, and this is the on my Mac. This is the version I have. You would need to get it also. So let's take a little break for 15 minutes now. It's 4:30. We will meet again at 4:45. But
Yes,
I am right here, and I want you to download and install Cytoscape on your machines. If you have any trouble, you can put a message in the chat box, and I will be very happy to help you. You don't need any. There's a DMG. I mean, there'll be a little tool. It's an open source executable, and you install it. And the moment it opens, it should open into some interface like this. And after the break, we will get back to understanding the interface better. Is that okay, Ruchi or Tabassum? Can
yes,
we stop here for 15 minutes now? You
yes,
can
ma'am.
stop recording if you're recording this session, and we will start again in 15
Yes,
minutes.
ma'am. Sure, sure. We'll meet at 4:45 now.
I'll probably put it in the chat box that we're going to have a little break. I'll put it in the chat box, the place where you can
It's very easy to download.
download
I
Cytoscape
already have
yourself.
downloaded.
What I would
No
love to
problems
have
at
in the
all.
meantime, while you're grabbing your tea and coffee and other good stuff, is if you put into the chat box a yes for having downloaded and installed. So if I have a yes from some of you at least, then I'll be glad that the next steps, which are going to be hands on practical exercises, can be done together. Oh, already downloaded. Very nice. Great. So now I'm going to let you grab a cup of tea or a cup of coffee, and then let's catch up again. Okay. What next? राम राम आप लैब में हैं क्या? All right. I hope people are starting to come back. And I see a series of yeses on the chat box, which means you have downloaded and installed Cytoscape latest version. Great. So if everybody has installed, and is everybody back, or should we give it a few more minutes to wait for
There are
anyone who
there are some
hasn't yet
participants who are
come
yet
back
to
from the
join.
break? Should we give it a few more minutes?
Yes, ma'am, please. Maybe two,
Very
three
good.
minutes. 2 minutes.
So let's get started. Let's get right back into the classroom.
Sure.
If all of you have imported or installed and downloaded Cytoscape, you will it will automatically open as you've moved it into applications, and you would open into this page. So here you have the Cytoscape interface. There are lots of different panels, as you will see on the page. Um, Here you will find some of these colored in orange, and these are the important panels that you need to remember. At the top is your menu, the main menu. The main menu is where you download and install apps. I would like you to do some downloading and installing of apps. So click on this little apps thingy. Wait, I'll put the laser pointer again. Here is the menu, Cytoscape apps menu. Click on the apps menu and you would find an app manager. Go into the app manager and press and type string, because we are going to be bringing out or using string app for downloading important genes, proteins, and pathways. So I want you to search and put um, the string app there. In my own case, and I've got my own cytoscape here somewhere. So this is my cytoscape. And on my cytoscape, here's app. I don't know if you can see the pull down menu. Yeah. And here at the top is app, my app manager. I don't have to go to my app manager because I have the string here. I have the string app. I also have the string enrichment app. You can get the whole suite of string apps by typing out string app. So if you go into the app manager, a little window will open for you like this, and there is a search button, and you can type string, and their string app comes in. See? So in my case, string app is already installed. In your case, possibly string app is not installed, and you can just press install, and that is, you'll be done in a couple of minutes. The other thing I want you to do is to download and install the wiki pathways. So you have the entire wiki pathways. Wiki Datascape would be a good thing too. I think I would like to install a Wikidata Explorer for Cytoscape. Well, this is wonderful. Anyway, so Wiki Pathways is what you need. I've already got it installed. You should get it installed as well. Okay. So let me see a yes or no on the chat box once you have installed these two packages. Yes, App Store is the one you need, Preeti. Very good. Thank you. So you get it from the App Store. At any cost, whether you have the app or not, your interface will look like this, whereby the top interface, the menu interface, is the one that I just told you about. You can take help from here. You can open new files. You can open old files. You can start creating a new network manually yourself and so on. Right below this menu is another little panel, and we call it the toolbar. And this toolbar is for you to see your entire network, in zoom in, zoom out, focus on something, search for something, and so on. Then on the left side, there's a big panel. It is empty right now on your new cytoscape. And this empty panel has a search window, which at the top is a little tiny window called the search window. This window which has a little left side area which allows you to search in specific repositories such as string, such as wiki, such as psychic. And we will learn all these today, the psychic as well as wiki as well as string. When you search for something in the world, so this is searching the whole world of data for anything that you might be interested in, your favorite species, your favorite gene, your favorite protein, and so on. When you search for it, a little pop-up window will come. It'll let you search, and when you choose okay, I'm searching for, let us say, let's say something to do with the circulatory system. And then you will get a window saying the circulatory system, we have information, published data for the circulatory system in Drosophila, in C. elegans, in humans. Where do you want to do? And then you might choose humans. And when you choose humans, then this little window, which is your network window. The network will be visible here. And when you click on the network, you will see it in the network view window, which is the main window of Cytoscape, where you already have a few example networks sitting here for you. Every network, the way you see it as nodes and edges, is actually a table at the back end, right? It is individual rows and columns of data. And those rows and columns of data, literally in an Excel style or a comma separated or a tab separated value style, are the back end of every piece of information. And those tables are your data tables, and the data tables are also visible in the table panel. The table panel is this niche wala panel here. So you could have a table of nodes. So when you click on node table, you will see only the nodes. Right now you see nothing because there are no nodes. You could click on edge table and you would see the edges. You could click on the network table. You could see how many networks are there, and so on. You could also have unassigned tables, which we will come to later. Okay. So for now, try and wrap your head around the interface of Cytoscape. And this is the basic interface of Cytoscape. Moving on from here, of course, the core concepts, as I've already told you, are the networks and the tables. So when you look at a network, you're basically seeing some nodes and some actors and some relationships, or nodes and their edges. But truly, A, B, C, D, and these blue colored lines are just visual representations of real data. So where is that real data? And that is there in the tables. So your annotations, your interactions, your relationships, your ties, your weights of edges are all in the data, and that is on the right side. Brilliant, Vijay Lakshmi Vijay. Interesting now. So very good. If you've downloaded the apps, that is, I will give you time later also. So those of you who haven't done it, don't worry about it. Kavyanjali says the app manager option is not available. That's not possible. The app manager option is the first option, Kavyanjali. How is it not available? Did you click on the apps? If you click on the apps, the first top option is the app manager option. Let me go back. How is it not? Is there somebody sitting next to you who can help you? Because here's the Cytoscape. When you go to apps, that's it. They're on the top. What's missing? What is it that you don't find available? What do you see there then?
It's showing app store.
Yeah, that's what you need to go to,
App
my dear.
store.
It's the same thing, Kavyanjali. Go to that one. Okay, maybe it's the new one. Man, it's a new download kiya hua hai, but anyway. Okay. So Krati says it is installed but not opening. Now, it could be true, which I've seen with some students, Krati, is that you're using a device that is not giving you permission for installing something new. If you're not the owner, then of course this we find with a lot of students at Cambridge also, that they've been given the laptops by the college and therefore you don't have permissions to install or open devices, open software. So maybe you can use one of your friends' ones, and when you have your own, you can use that one also. So this is it. Click on App Manager and you get the app. And let's come right back to the. Oh! I didn't want to close it. हो गया बंद कोई बात नहीं we can reopen it another time. So the core concepts of Cytoscape are its networks and tables. Yeah, so my Cytoscape is 391, whereas yours is 310.3, and that's why you guys are seeing this. So yours is better. So go to the App Store and get the stuff. That's not a problem. Um, the core concept, as I was talking about earlier also, is that of networks, and at the back end the real data of the networks is in the tables, and this is how a table would look. But if it's very very simple, you would just have the name of the node, and that would be all that you have. Okay, you can improve the basic network visualization is this blue and pink color. This is the default. This is okay, but you can improve it if you learn how to use networks, how to build and visualize networks, and how to use the different features of Cytoscape. Your network could actually look something more interesting than the default network. In either way, it will still be representing exactly the same table that you had earlier. So this table can be represented like this, or what I showed you on the previous slide as blue and pink, or something else which you and your curiosity driven brain or your creativity can allow you to change the visual. So visual styles are changeable. Topology can be modified as you want it to be visible. Okay, and that is something that we will learn today. The first bit that you need to understand is, where do you load your data from? How do you load your data? Okay, so in the loading network data, oh, there's a question by Palak. Does anybody want to answer it? Those of you who have done it already, please answer the question for Palak. It's a very simple answer. Palak, we are asking you to download 2 specific apps. Some of your friends will tell you. Otherwise, we'll come to a slide. Now the slide is coming. You can take that also. Okay.
There's App Store.
So you can load the network data as different kinds of files. Maybe your data is sitting in an Excel sheet on your computer. Maybe it is sitting as a TSV, CSV. Maybe you have some more complex formats such as the BioBax or SBML or GML, or you have some very simple formats like simple interaction format that I will teach you today. You can load these from, or you might say, okay, I can't load a network because I don't have a network on my machine. I have nothing. I just want it from the public databases. So there are zillions of public databases, as I taught you today, where you can get the data from. So some of these public repositories from which you can download the data are Psychic, String, Intact, Reactome, Wiki Pathways. And this is the answer to the question in the chat box. Palak has asked the question. Ilma has also asked the question. The answer is right here, my dear. So you go to the App Store or the App Manager, and then you get the String app. This is the name of the app. Type it out in the search box, String app, and you download the String app right there and get it. Or you can also, I told you to download the Wiki Pathways as well. There are 2, 3 more that you can do. One is the Intact app. So if you have the time and if you are interested right now, you could just search and install Intact app also, and the Reactome F1 app also, so that the reaction chemistries are also available to you to choose from. And the Intact is the largest primary database for protein-protein interactions. So all the interactions in the world are available at Intact, and you could have, straight away you could have access to the entire known universe of interactions between proteins in biology that have been published at your fingertips. The other thing you can download is Pathway Commons through the SciPath 2 app, but these are completely your choices. What I'm going to teach you today in the next couple of slides and the next couple of, in the next couple of exercises is to use the String app and to use the Wiki Pathways app and maybe the Intact app. Okay. And much of the automation of all of that you do in Cytoscape can be performed using command line scripts or Python or even within R, but you don't have to know that today. You don't have to have any coding background at all. All of this you will need when you are you are repeating the same process thousands and tens of thousands of times. Only then do you need a loop and the code. But if you're doing it one time, if you have a single interest question in your mind, such as ovarian cancer, let us say, or leukemia, let us say, or floral transition, let us say, or as in my case, plant communication, let us say, or secondary metabolism in medicinal plants and so on. So if you have a specific question and if you have a specific research interest, you could straight away start the hit the ground rolling and put that question, and you would get the answer, and no need for automation of that for over a zillion other iterations. Okay, so loading the tables can be done, as I said, from files or public repositories or automated through scripts. And we will now be loading our first network. So to load our first network, you should have your Cytoscape window open. You should have your internet on because you can have access directly to different public databases. So in Cytoscape, and these networks are from the Cytoscape tutorials. I hope many of you have noticed that many of my slides are originally from the Cytoscape tutorial data or collected from various parts of the web where I've tried to put the source where I got those slides and those pieces of information from whenever they were useful. I've put the source there. You can get more information at the sources or the papers or the links that I have provided. In Cytoscape, network data can be loaded from a variety of different sources and in several different formats. Where you get your data depends upon what kind of biological question you have and what kind of analysis are you planning. Right now, this tutorial that we are going to do is going to tell you how to load network from several popular sources in several popular formats. Okay. So the public databases that you will need here are index, psychic, string, wiki. And you don't need the other local and remote files. So here, this is what all of you have done already. Go to the Cytoscape menu, pull down the menu under apps, and then choose the app store or the app manager. Once the app store or the app manager opens, a little small window will come. Then you choose wiki pathways. In my case, it is already installed. In your case, if it is installed, good. If it is not installed, you should install it by pressing the install button. That is the first app that I want you to install, which is the wiki. And the second app that I want you to install is the string app. And the string app, you just type string into the search box and you will get the string app. Can I get a show of hands from everyone? Can I get a message in case somebody has not been able to do this? If you've not been able to do this, then I'll wait a couple of seconds and make sure that we are all on the same page before we go ahead. And I can give it a couple. Give it a 30. Thank you, Chavi, but this question is for those who have not been able to do. So if you've not been able to do, maybe I can ask somebody to help you or I can help you myself if you've not done it. So I'll wait for another 30, 10, 20,
Divya,
30 seconds maybe to see if there's anyone who's been left
Yes,
behind in
ma'am.
the class.
you have raised your hands.
Sometimes those of
Hand.
you who have not been
Ma'am,
able
I have
to
downloaded it.
All right.
get there forget to ask or are a little embarrassed to ask. Don't feel embarrassed here. I need you to be, yeah, thank you, Tista. That's the answer to your question, Krati. Wiki Pathways, String app, very important. But Intact and Reactome F1 plugin is completely up to you. Great. So the apps are named there already, Krati. I hope it helps you. Wiki Pathways, agar aap kar rahe ho, to that is good enough. No, that's good enough, Tista. Brilliant. Okay. So if you have correctly installed all the apps, then when you do the search, when you go to the network search window, all of these options should be available to you in your search query. Are they available when you press down the network search window? Do you remember the network search window which I showed you on the interface of Cytoscape earlier? Let's go back and I can show it to you again if you have forgotten. It was here. I think it was here. This is the network search window. This one. You see this little guy? You see this little arrow? Press it down and a whole menu should open up. And out of that whole menu, don't do anything till I tell you what to do. So click here in the network search. This is the search panel. Click the search panel and do as I tell you, but you should have the option to search among all of these different places, which are Psychic, String, Intact, Wiki Pathways, and so on. Here. So does everybody get these? Once you have these, the resources, these are the resources where you will make the search. And the resources will depend upon what apps did you install. Okay, today I asked you to install these string app, and the wiki pathways. But if you have installed more, you might have even more. So for now, this is great and good enough. I want you to, yeah, let's search for the P53. Everybody's heard of the P53, which is a major cancer biomarker. And so put the transcript of P53 or just P53 into your search box. I have tried to make it even shorter by making TP53 and BARD1. So we've put both of these guys here, but put that into the search for index. Just search the index. Do not search Psychic yet. Do not search String App yet. Just search the index for TP53 and BARD1. You will get a lot of options. Now here what you see are the outputs that I found when I first started teaching this course. And these are very basic outputs. So BARD signaling events are there. There are many networks that you get as an output, and your choice will have to be, you will have to choose one of these, and then you will be able to load them into your cytoscape. Why don't we do it together? I'm going to try, and so you can select one or more networks to import, and you can click on the import button to the left. Why don't we do it together? Let's try with TP53 and BARD1 together. Okay, where is my cytoscape? Here's my cytoscape, and there's a new version. I don't want it. I want to go to my networks and I want to search index. So I'm going to search index for TP53. What happens? Yeah, it takes time. It takes time. So I'm glad we are all not sitting in the same room, because when we last did this hands-on tutorial here at NIPGR with the PhD students, 20 students or 25 students started searching at the same time on the same bandwidth in the same room, and some of them completely got knocked out of the system. So because we are all online and in different places, I'm sure that you will be able to search um, the oh, sorry, I'm searching wiki pathways. Sorry, wrong. Cancel this. You're not supposed to be searching wiki pathways. You're supposed to search index. My mistake. You got to search index. Remember? That's what you got to do. You got to search index. So, has anybody found what you got on index? Okay, right here. Oh, band ho ja sim. This one won't close till I. Okay, I'm gonna quit it because it started searching. I'm gonna get inside Cytoscape. That was a lot of search. That is why it's important to put TP53 and BARD1 both, because if you put one, you'll get so much more data. So it's important to limit your data in some way and see what you can get. Okay, let's see again. Close. Index, TP53 and BARD1. Search. Has it taken a lot of time for you to get it? Oh no, it took 5 seconds. It got me this answer. Has everybody got this? Okay, so BARD1 signaling events are public data, and there's 29 nodes and 180 edges. Let's try to look at this one. And this, when you hover your mouse over the BARD1 signaling events, you find that this is a list of entities from the original network that has come from the Indra database. Okay, it's a database that integrates information from multiple spaces. And let's say if I choose EGFR, this network, the EGFR, is a protein-protein interaction wild type using the luciferase-based bioluminescence resonance energy transfer experiment. And the data is also public, of course, and there are 341 nodes. So I would suggest, because we are all doing a little experimental exercise, let's not go for nodes, for networks that have many nodes. Let's choose a smaller network, such as the BARD1 signaling event. So let's click on the green button here, and let's import this. So this is now getting imported into my cytoscape, and I would like you to import this network into your cytoscape as well. So I'm going to close this network because it's already imported into my cytoscape. Has it been imported into yours? There it is. Do you see this little network that I have at the side? It's too tiny. I want to see it bigger. If you want to see it bigger, you just go to the little window where I was teaching you, you can do a zoom in or a zoom out. Choose this one where you do a fit content, right? The third symbol here is fit content, and there you get reds and blues, and that was very much like what we saw here. So the barca 1 is there, the atr gene is there, the fancy f gene is there, the rad 51 is there, many common genes that we all already know to be associated with the TP53 are already on this network, and which is what you all also got. So once this network has been imported into your network, Psychic will do a different game altogether. Now we have up till now we were searching index, but now I would like you to search Psychic. Okay, Psychic contains data from multiple sources and multiple types, including internally curated, experimentally observed, and predicted data as well. The Psychic web services can be easily accessed in Cytoscape as a core app. To search Psychic, select Psychic from the network search and then type in your search term. Let's try TP53 and let's see what we get. When you search Psychic for TP53, you will find that there are lots of different kinds of databases. But the interesting thing that is different between Psychic and Index is that Psychic allows you to combine all of these networks and take the common genes. It could be that Intact has 873 records, whereas the Reactome FI has 300 records and the Matrix DB has 50 records. But you want to make sure that you're not missing anything because you're doing research. You're not doing, in general, when you're trying to address a project, you want to know what maximally has been known about that particular gene, that particular protein, maybe that particular pathway of interest of yours. And therefore, Psychic is where you can get access to multiple different data sources to choose from and combine. So here what we will do is we are going to choose the Intact and the Reactome FI outputs. When I did this as an example, and I can show you, and what you're seeing on the screen right now is when the last time I was doing this, Intact TP53 data had 873 records, whereas the Reactome FY records for TP53 were 369. Can somebody tell me what it is today in your search? When you search the index for TP53, do you find more or do you find less records in Intact and Reactome FI? I'm waiting and looking at the chat box to see if, okay, Palak says that Intact as on date has 1,271. Very good. What about Reactome F1? Reactome has 369. Okay, it's the same as what we had earlier. Okay, very good. So you can just put a tick mark in both of these guys. And okay, depending upon your internet speeds, if your internet speeds are bad, don't do it. Try any other two. But if your internet speeds are good, then you can click on both. But remember, when you click on both, two networks will download. And earlier you have already downloaded that other network. Remember the index network for TP53 and BARD1? That is already one network. Now these are two more networks. You don't want to increase the number of networks that you have in your Cytoscape interface window. And so therefore, what I will suggest is that you use network merge. When you use network merge, then you will only use the common bits in the two networks. So maybe Intact has some records which are common to Reactome F1. Maybe Reactome has some records which are already present in Intact. And therefore, you will not have a doubling of the data or the repeat occurrence of the same records by clicking on automatic network merge. So at the bottom of this page, you have the option to automatic network merge, which you should do for Intact and Reactome. And in principle, I don't want you to do it now. In principle, when you're really doing some research on some question of interest to yourself, you should click on all. I mean, you should have time and you should have the space and you should have the storage space and you should have the bandwidth, and you can click all and you can merge the network so that all the data that is available comes in. Divya's intact is giving 1400, whereas Ashia's intact is giving her 1200. Very interesting, Divya. You seem to have a newer version of intact than most other people. Very nice. So my intact, of course, has 873 only. So we're going to take both of these and we're going to merge the networks and we're going to press import. When you press import, this network is going to come in to your um, um, uh, cytoscape and get imported as this. You are importing interactions from Psychic, you're loading the data, this is how it will look, and it will be as many tasks, as many networks as you pressed, it will take time. So wait for it, give it some time. You will get this window which says importing the interactions from the Psychic services, loading the network data from remote Psychic services, and it will slowly come in and it will give you okay, Ilma, that number is a bit on the crazy side. I don't expect millions of interactions. Maybe you didn't put the same search as others among us. I don't expect 1293542, my dear. Anyway, coming back, when you and don't import such a large number, I don't know what search you gave, maybe all of the world's interactions came in. So importing the interactions, because you have chosen two different networks, it will be 1 by 2 task and then there's 2 by 2 tasks. And you wait for these to finish. Okay. In my case, with the download is still going on and it is still waiting for the download to happen from the intact database, whereas the Reactome, because it was smaller number, Reactome came faster. You all also do this and see how long it takes. And the moment you have them downloaded, your network window will have the new network created here, the name of the network, and on the right side you will have the visualization nodes and edges. And at the bottom in the table window, you might even see your table, the actual data table. So let's see how that happens. This is how it happens. So the merged network comes in as a combination of interaction data from Intact and Reactome. And Psychic also includes data from many species. And these many species are available in different colors if sufficiently represented. Right now, who's going to tell me how many species there are in this data? Let's do this together. Uh, all right. P53 search किया आपने. All right, let's try P53 on Psychic. When you try P53 on Psychic, you get 1490 on Imtact and 201 on Uniprot. And then when you download it, you get something like this. The P53 gives you a network. On the left side, you find the network written like this. It has, who's going to tell me how many nodes are there and how many edges are there in the downloaded, imported, and merged network? It's a big merged network. Who's going to tell me how many nodes and how many edges on your network, whatever you downloaded? And it's very easy to see because it can be read off. You don't have to count one by one. You can easily read it off on the network window right here. See the merged network? It has 872 nodes in my case and 920 edges between these 872 nodes. Does anybody else also, can you read off your network nodes and edges and tell me how many nodes and how many edges your network has? Also, you should put the search criteria that you had given. So in my case, for example, I gave P53 as the search term. And for the search term, I got two networks that I downloaded, Intact and Reactome. And from there, when I merged them, I got 872 nodes and 920 edges. Krati has got 900 nodes and 9 edges. Not possible, Krati. That is not possible. 9 is too less. Usually the number of edges are more than the number of nodes because all the nodes, if there are no edges, then those nodes will not be connected, right? So anybody else who will put the answer? I'm going to put my answer into the chat box. And my answer is that I gave the search of P53 and my output was 872 nodes, as I can see here, and 920 edges. So that is my that is what I get. In the same way, I would like you to put in the values for what you have got. And this is only I'm asking you to do so that I know that some of you are doing this. It's important that you do the hands-on exercise sat so that you can understand the later exercises, which are a little more, where you will actually start doing network analysis. Ilma, it's not downloading because you chose something like millions of this. You need to choose some of the smaller ones. So that's why I'm asking you to put in the number of what is the search term that you gave, and based on the search term, what is it that you downloaded and how many you downloaded. If it's not merging, maybe you've chosen something very, very big. So choose something smaller, go back and choose something smaller, no worries. If there's an error, don't worry about it, restart it. Don't worry about it. Sometimes we get errors, no worries. And in fact, I will, do it on um, my cytoscape myself so that some of you who are having error can see how I'm addressing those errors. Okay. So here was initially I had searched TP53 and Bard1 on index. All that was very good. I got a network. All that is very good. And then I searched on Psychic. On Psychic, I made a search for P53, just P53. So you must make sure that you've typed out the correct term. This is P53. And when I put a search for P53 on Psychic is searching for many, it's given me many, many databases. So, Ilma, no kind of search for P53 will bring back millions of records as in your case. So, I think you did not, what I believe you made a mistake with was that you did not put the correct term in the search box. So put the correct term in the search box, whatever you're interested in, and then you will find these records here. And then you choose something. So I told you to choose Intact and Uniprot, but Intact and Uniprot have 2000 and 200. I want to do it faster because this is just an example. So I'm going to choose HPIDB with 26 records and Uniprot with 289 records. And then I'm going to automatic press merge. So you have to press this merge button. That's why it is not going to bring it. Yeah, Intact is 2053, Ilma. That is correct. Absolutely. This is what mine also shows. See? So if you follow me the way I'm doing it here, you will also get the same records. Now, I want you to choose any two of these because we are learning how to merge different databases. One is the HPIDB, which is the protein internally curated mimics curation with evidence. And the second one is Uniprot, which is another protein database, which has also nucleic acid protein database and small molecule protein data, as well as internal curation, IMEX curation with multiple evidences. So it is one of the most famous databases, Uniprot. Now we take both of these, click on them, and press automatic network merge. Press the automatic network merge and then say import. Okay, press import and it'll take some time and boom, it's there. Within minutes, I got it. You see what I get? So, what do you want to do now? Networks created from these databases. I don't want to do anything. I want to close. So, I've closed it. Okay. So, what is the error you're getting? Preeti, see, when you're in a group, no, when you're trying to tell error, showing error is not the right thing. You should say, what is the error? So, maybe the error can be addressed. Oh, there's a Java error. Oh, if you're having a Java error, it basically, I got it. All of you who are having the Java error do not have Java installed on your machines. Okay. If you don't have Java on your machine, then it won't run now. So you need to install Java, latest version of Java on your device, and only then will this work. So don't worry about it. Don't do it. If you have a friend with you who's for whom it's working, look at theirs. Otherwise, just look at this screen and you can come back to this recording and you can play it next time after your Java is installed. That's an important feature. I didn't realize that so many of you will not have Java on your devices. So this is something I could put as a feedback into my first slide where I ask you to download Cytoscape and the apps. I will probably also ask you to check whether your computer has Java and the
Um,
latest version of Java. And if it
Gitanjali,
does, then it will. So
actually,
without Java, it will not search and bring it out. No worries, no worries. Good
Gitanjali,
points. So
actually,
here,
when
just keep
we
looking at my screen. In my screen, do you see multiple networks that have come as the output of this? And so of the search for P53 and my network. Can you see my screen? Can you answer the question of how many nodes are there in this network and how many edges are there in this network? Of course, it's written here, but it's too small for some of you to read. I hope you can read it. And if somebody can read it, please tell me the number of nodes and edges in my network, which I have downloaded. If you are unable to download, if you are able to download your network, brilliant. But Bhumi is absolutely right. 172 nodes are there and 169 edges are there in my network. Okay. And here is the table. So this is the backend table which I want you to focus on. So if you click on any one of these nodes or genes and go to the node that is the single guy you've clicked on, and that is the information on this single gene. Its name is Q9UNR9. It's from the human Homo sapiens. And what else do we know about this guy? I don't see we know much about this guy except its name and it's the fact that it's from humans. Let's click on Q9UB, this central guy. This is also a human protein. This is a tumor suppressor P50. Of course, that's the P53. This is a human protein too. So it looks like, yeah, P53 is all about humans. So we find everything. Oh, there is a different species. Here I find a different species. This is Gallus herpes virus or Gallid herpes virus, which also has the P53. This is the Drosophila melanogaster. This is also Drosophila melanogaster. This is, I think, Chlamydomonas something. I don't know the full form, but we can find it out from this taxonomy ID of 9534. Here you again have Drosophila melanogaster. So yeah, few different kinds of species that we find in this. Very good. So now I would like you to know how to modify this network. So we have this network, or maybe if you earlier got the BARD1 network, we still have the BARD1 network also, and I can make it bigger so it becomes visible to all of you. And let's say I decide suddenly that I don't like the way the network looks. I want to change its network topology. I want to change its style. So what I can do is I can go to network styles, and here there are so many different features about the network that you can change. You can change the paint, that means the colors, the widths of the borders, the fill colors, the height of the node, the edge size, the font size, the position of the label, the shape of the node is circular, but you could change it to anything else. You could choose hexagon, you could choose parallelogram, you could choose a rectangle, and so on. So if you choose, let us say, a triangle and applied it, all the nodes would change to that one. If you didn't like the fill color, and you could have a complete color brewer in front of you, and you could choose from any of the other colors that you like. For example, if I like purple more, I can get all of my nodes to look purple. Okay. And the label color can be made something else. The border paint can be made something else, and so on. But sometimes changing each one of those features is a lot of waste of time unless you're trying to publish something and you want to show something specific. So what we will do is we will just choose some of the general styles. What are the general styles here? So if you go into style and if you search with properties, uh-huh, where's it gone? Create a new style, refresh a style, make this a default style here. When you pull this menu down, you will see that right now I'm using the barred signaling event, this purple color that I've created, but there are other mechanisms in here, curved, let us say. If you put curved, your network layout completely changes. If you don't like curved, you might choose a dark background because you've got a black, let us say you like the string reactions better. So you could put this, but the fill color is silver and you don't like silver. Let's say you want them to look a little bit different. You could change those colors and you could make a different color in your fill, and those are the label colors. Oh, that's the label color. Fill color, if you want to change, let's see what fill colors we might be able to change over here. Try a few more playing around with different kinds of nodes. So oh, I've still not been able to change these colors. I hope you have been able to. So you can play around with all this. Maybe you don't like the black background. You want to have something white background. So that's a very simple layout. Whatever connects with you, whatever makes you feel better about looking at something. Maybe I do like this layout, but I don't like the background. The background is too dark. So I can just choose the background myself. Here's the background paint color in this style, and it is brown. Let's say I don't like brown. I want it to be very, very lighter shade of white, let us say. So I'm going to go back to white and I'm going to say, okay, try and make my background white. The moment I make my background white, all of the edges disappear. So I come back and I stand corrected that maybe it was not such a good idea to make my background white. So I make my background colored. But the moment I do that, I realize that the labels have gone disappeared. And so maybe it's a good idea to stick with the default that it was, where I could see whatever I was seeing. Okay. So try different networks, try different visualizations, play around a little bit, fool around a little bit. If you destroy something, it'll still come back when you log in next time, and so on. Okay, so this is how you play with the layouts, and you change the styles, you change the colors, you play around with nodes, edges, or the whole thing, whatever you like, change whatever you like, and do what you want to. There's a lot more you can do here, but we will see about that later. For now, we are going to move on in the little master class on these networks. And we are going to say, you can, okay, I've taught you style already. Check how these colors are assigned by Cytoscape. By going into style, you can change the fill color of the nodes. You can change the height and labels of the nodes. You can choose what other colors you want. And you can give specific colors to specific nodes also. Okay. And this can be visible in the node table. When you go to the node table, which is your little tabular window below, you can see what nodes have been given what color to. So here, for example, if you click on the node table, you will find that there are human genes. We also find rat genes, as you can see on this slide. So this is not entirely human, the network. If you only wanted human data, then you can put a search, a new search within this network here with this let me try the laser pointer again. You can search, there is another search window which is here. The first search window which I told you for is to search the whole world for your network. But now you have your network, you can search within your network for only human data. So you will click human here or type human here, and you will only get the human data sets. And then you can build a new network with the human data set so that you have the P53 human data. Okay, now we're going to load a network using string. And I hope everybody has installed string because this is the next thing we're going to do today. And to search the string, select the string disease query in the drop down menu at the top of the network and choose a disease. We are going to choose a disease now. And because I was talking about ovarian cancer earlier, let's choose ovarian cancer. Of course, you can search for any disease that is of your choice. The resulting network is going to automatically load. So here in the pull down menu on the search box where you had earlier chosen, what was it? Index, and then you had chosen psychic. This time I want you to choose the string disease query. Okay. Because you have already downloaded the string app. So you have the string disease query. And with the string disease query, type ovarian cancer. When you type ovarian cancer, all of the data in the string disease database gets downloaded here and you find whatever is known about ovarian cancer. So for example, when I did this search and I put it on the slides, these many, like how many could there be? About 100, 150 genes or proteins are known in ovarian cancer. And these are also interacting with each other, like you can see on the slide. And this is the ovarian cancer data. This network is going to load automatically. And you see how it's different from the previous network. The previous network was very monotonously colored. But string, because the string app, I don't know how many of you have used string app, but a lot of scholars prefer the string app because there are different colors and structures are visible also. Within the string, if there is a PDB structure or if there is the availability of a three-dimensional structure for any of the proteins that are there on your network, they would be visible. Okay, so let's try ovarian cancer, and I would like you to tell me how many, at least those of you, even if you don't have Java, when you press ovarian cancer, you would immediately see how many different ovarian cancer genes and proteins are there in the string disease app. Okay, so I would like you to tell me whether your ovarian cancer data looks like the one that I'm showing you on the screen, because this was done a long time before today's class. Okay, so I'll wait on the chat box if some of you can put information about the ovarian cancer. It should look like this. And you can also use string specific styles. Okay, you have 99 nodes and 2000 edges. Brilliant. So when your network loads with a string specific style, that style is unique to string because it includes, as I said already, three-dimensional protein structure diagrams within the node. Okay, so the string networks also include data as the node interaction attributes, which can be used to create a new style. So the string app includes options to to change the interaction confidence and expand the network. Now, you see this carefully. When you're downloading data from string, it will always thank you, Vijaykash Mukherjee, 992301. That means as on date today, it is 99230. Brilliant. So now what I want you to do is I want you to modify this network, change the confidence level in string because string is a predictive. Remember when I was teaching you about primary databases earlier today and metadatabases and predictive databases? What was string? String is a predictive database, which means that some of the nodes and some of the edges that you are getting here are predictive, which means there is a confidence associated. They're not facts, they're predictions also. And so, therefore, it is important for you sometimes to be aware that you're doing research, and it is important to begin with the facts, with the stuff that is known at a high confidence level. And so, therefore, you have the option in string to change the interaction confidence to a certain level and then expand the network or shrink the network based on that information. So let's try it ourselves today. Um, oh, okay. Okay, so you can do that. But what we will do is we are going to move on to the third type of loading the data. Okay. And the third is the pathways because I asked you to download and install the Wiki Pathways app also. We are going to now what we have seen is individual genes and proteins can be searched on index. Individual genes and proteins can be searched on psychic. Diseases can be searched specifically on the string disease query. And now we are going to search for a fourth kind of search because some of you may not be interested in a disease. Some of you may not be interested in a gene or in a protein. You may be interested in a particular pathway. Now, I'm working with the mevalonic acid pathway or maybe the non mevalonic acid pathway. You might be working with the statin pathway. So let's go to search the Wiki Pathways and let's select because Wiki Pathways is a collaborative wiki platform with manually curated pathway models, and it currently covers more than 2,500 pathways, and it has very species specific, it has collections that are species specific. So you can say that I'm interested in the statin pathway for humans or statin pathway for rats or statin pathway for ferrets or statin or a certain pathway in plants or the floral transition in Arabidopsis and so on. Okay, so let's search Wiki Pathways. For those of you who have downloaded the Wiki Pathways, make sure that your network search points to Wiki Pathways and then you type out the word statin. You're going to select statin and then press the search button. And I want you to tell me what you get as a result. When you press the statin search, you would get a series of statin pathway data sets within the Wiki Pathways. One will be from buzz, like the mouse, then there is the cow, there is the carrots, there's the rat, there's humans, there's also the Pantroglodites. So, choosing or based on your favorite species, which you had put for me initially in the chat box, you can now choose to download the statin pathway for any specific favorite species of yours, such as the Rattus norvegicus, for example, and you can import it number in two ways. You can import it as a pathway or you could import it as a network. Now, there is a difference between these two imports, and we are going to see how the pathway model opens up when you open the pathway as a pathway. The default is the pathway import. But if you want to import the network as the pathway as a network, then you have to click on import as network. Okay. So does everybody get this? And you can choose which one you want. But if you want to click on exactly what is visible on the screen, choose the statin pathway and choose Rattus norvegicus. And what do you get? What you get is here downloaded in both ways as a network as well as a pathway. Brilliant, Krati. I'm very glad to see. So you already have 48 nodes and you already have 26 edges for the statin network in the rat. And the left side visualization is that of a pathway, which means the way we are used to studying in Leninger, right, in college and in kindergarten. When you study a pathway, it is visible like a proper pathway. But when you visualize the same pathway as a network, on the right side, you will see it downloaded and visualized as a network. And that is more important because the network visualization is what I need you to wrap your head around. And that is what you're going to learn and try and improve with time in the way you represent these things. So the Wiki Pathways model of statin is represented and imported as a pathway in this page as well as a network. Maybe your pathway and your network will look a little bit different from the one I am showing here because this was done some time back. Now, does anybody know how many different cellular locations the pathway statin pathway is existing in? One, as you can see here clearly, is the liver cell. Then the pathway is also in the plasma. Some of it is also connected to the cholesterol. And then, of course, on the outside, you have the cell membrane. And it's very interesting if you map both of these together, you know that the normal network visualization does not um, let you see the spatial location like the liver cell or cell membrane, but the tables already have this data. And so you can always put a color on the nodes in such a way that you can see all the nodes from a certain location in one color and all the nodes from another location in another color. So Nayan, very good, good answer. And if we move on from here, okay, there's a little challenge now. We are coming towards the end of today's lecture, and I would like you to do this challenge yourself. I'm going to wait for 5 minutes for you to do this. You don't have to complete all of it, but even if you are able to do a little bit, please put a yes in the chat box to say that you got it. So you have to put the answer. Well, you have to basically perform a single search using the protein called Notch3, and then you try it with the gene symbol Notch3, which is NOTCH3, and then the UniProt ID of the same Notch3 protein, which is Q9UM47. Perform both the searches independently. First, you search with Notch3, then you do with Q947. And then tell me how many interactions did you find when you searched for Notch3 and how many interactions did you find when you searched for Q9UM7. Okay? And you're going to search this over index, sabse simple wala, index. So, how many interactions did you find in Notch3 for Q9? And then you can do the psychic. Within psychic, you will find that intact has some records and IMEX has some records, and you can tell us those numbers. And I want these numbers as answers in the chat box after you guys have done. Okay. And also, the last question, which is the most important one, is when you search for Notch3 and you get the network, what is it different from the network that you get for the same Notch3 when you use its Uniprot account ID, which is Q9UM47? Actually, both are the same Notch3, right? So you should get the same network. But this exercise is to show you that maybe they don't look the same. And then we can have a little philosophical discussion on why they don't look the same. Or maybe I want you to think over it overnight and then we will have that discussion tomorrow. Anyway, this is your homework. Well, it is not your homework, it's your current challenge. You're going to do it now. I'll give you homework later. So we have 5 minutes. It's 17:40. I'm going to wait till 17:45, grab some water myself, and look at the chat box to see if any of you have been able to do this. Okay, your time starts now. Oh, very good. Preeti has an answer. No, not just Preeti. Many of you have an answer. I'm glad to see that. Okay, so for notch 3, you find 92 nodes and 212 interactions. What about the same name for the Uniprot AC, which is Q9UM47? What do you get? The same numbers, less or more? It looks like less because I can see the answer given by Palak. No, Palak, you've put notch 3 and notch 3. What is this? Um, I was saying try it with notch 3 and then with Unipro with Q9UM47. Okay. Okay.
I don't know. I
Notch
have
3 gives you
committed.
54 and 78. Okay. That's
I
what
have.
Amarjeet says. And then when you put the Uniprot ID, you get more or less, is the question. I'm realizing that you are somehow getting more with Uniprot. Yes, Preeti is right. I'm surprised that you're getting more with Uniprot. That is difficult to believe. I think you should get less with Uniprot. But anyway, I'm not going to question your output. Preeti, can you put your numbers on the chat box and tell us what you get for Notch 3 and what you get for QMUM47? Mm-hmm. That's the Q9. Yeah. So I want everybody to look at Preeti's answer here because that one looks to me like the correct one. Notch 3 is a is a how should I put this? It's a synonym. It's a name. It's a gene common name that we have given to the Q9UM47 Uniprot ID. And when you search for the common name, you will end up finding some extra information, which may not be related really to the protein, which is Q9UM47. Q9UM47 is a very specific ID and it will give you information exactly for that specific ID. So you would get it less. But it seems only Preeti has got this output. Everybody else is getting very different numbers. Maybe what we are doing is we are searching for different, we are searching on different databases. Are you searching within Index or Psychic? Are you searching the Psychic? Within Psychic, you get the option of searching the different databases such as IMEX, such as Reactome, such as Intact, and so on. And that is what maybe you should have done anyway. But I'm very glad to see that a lot of you are trying a lot of different things and you are getting very good answers. Let's see what I have on the next few. So 5 minutes are over. We are moving on. This is the output. Okay, so the numbers may be slightly different, but when you do a search for notch 3, you in general you find IREF index has 459 records. Whereas when you search the Q9UM47, IREF index has only 402 records. When you search for notch 3, the common name, your intact has 43 records. And when you search for Q94M7, intact has 42 records. So basically, Q9UM47 is giving lesser result than notch 3. Can somebody tell me why this is so? Why the intact ID is going to give you a lower number than notch 3? And this is how the network looks also. When you download the network, merge and download the network, the notch 3 network has 299 nodes and 305 edges, at least in my case. And of course, these can be different in yours. Don't worry about that. But the Q9UM47 has 263 nodes and 269 edges, less, as Preeti has said already, compared to the notch 3. And the reason is that when you search for a common name, quite often we use, I'm interested in P53, I'm interested in Barca 4, I'm interested in Barca 1, or I'm a plant scientist, I'm interested in floral transition, and I'm interested in leafy. When you put words which are common names such as leafy, notch 3, Barca 1, TP, you find a lot of random hits for that English word coming in the entire cosmic universe of interactions that are published. So, what you end up getting if you're a researcher, you're going to get false positives. And therefore, as a scientific researcher or a biologist who already knows the correct scientific name of your gene or your protein which you're interested in, I would say that you should search for that name, because then you get the number which is exactly of interest to you. अब देखो, the upper network has more clusters. It has some small cluster here, some small cluster here, some small cluster which is completely detached lying somewhere else. It's because that is a notch 3 in some other place, some other thing of complete irrelevance to the Q9UM47, which is your biological question. But if you were looking at common name and searching by common name, you would go back to your teacher or your supervisor and you would show this network, the upper network, which will have false positives. Therefore, as a scientist, as a researcher, as a scholar, it's very important that you search for the correct ID. First, you find the correct ID for your protein or your gene, and then you start making that search. All right? So that's the lesson, that's the moral of the story. And here is the example also. When you do the notch 3, which is actually Q9QM47, you get exactly the same kind of network, except these side two unnecessary, irrelevant, non-connected P78745 type network, which was not here in Q9QM47, which means you got the correct network by studying this guy. Okay, I hope that I hope that addresses the doubts that some of you might have had. So anyway, that's it. Now I'm giving you some homework for tomorrow, and this homework is that you're going to perform a search using leukemia. No, first of all, you're going to get Java loaded on your cytoscape so that tomorrow we can do more exercises and we can understand network partitioning as well, and we can do it together. Okay. So the homework is for you to perform a search for leukemia in the string disease database. Now, leukemia is not to be searched on the index. Leukemia is not to be searched on the where on the psychic, you're going to search leukemia in the string disease database where we were looking at the ovarian cancer. And make sure that you don't download and import everything that the network gives you. You have to import only at network confidence zero point 0.7, and the maximum interactors you can have is 250 because वो download करना आसान होगा. And then there are 4 questions that you're going to answer. And the 4 questions that you're going to answer are: how many nodes and edges are there in the network? How many genes are connected to FLT3? Which gene has the highest expression in the bone marrow and the gallbladder? And which term has the lowest FDR value according to Wiki Pathways and Uniprot? Oh, this looks like a tough bit of quite a few questions, which I think I should help you through. So let me see if this homework can be given, or should we have 10 minutes. We can do the answers together and I'll give you some other homework. Let's do that. Let's do this together only. So, did you perform leukemia search in the string disease database? How many nodes and edges did you get? Here is the answer. Perform a disease search for leukemia in the disease database, and you find that the leukemia, you have 4 outputs on the disease related to leukemia. There's the leukemia myelocytic acute leukemia, then there's the myelogenoid leukemia, and then there's the leukemia of the central nervous system. But what I want you to define is only the leukemia. And when you search for leukemia, string disease will give you the option to search for a certain confidence level where you have to choose confidence point 7 and a maximum number of proteins. So you could choose thousands of proteins if they are there. Okay. So leukemia, if you keep on going, it will keep expanding the network. String allows you to expand the network, and therefore there's no number. You can start with the basic core of 10 genes and then who they interact with, then who those interact with, and you can keep expanding the cosmic universe as far as you like. But we're going to put a border and a cutoff, and you're going to search for leukemia now with zero point 0.7 confidence and 250 maximum interactions or maximum nodes. And then you press, oh, you have to get the enrichment data. You need the enrichment data. Enrichment means any kind of additional metadata about leukemia. And that is where you will find gallbladder, bone marrow information for that disease. Okay. If you don't press enrichment data, then a lot of additional information that is already there in the databases will not be downloaded for you. So click this and click this and click this and click that and then press import. Anybody done it? What did you get? Brilliant, Palak. Brilliant. 248 and 3969. At the confidence? No, Palak, that is wrong. See, if you had put the maximum number of interactions at 250, then how you can get 3069? Oh, maybe you're right. Maybe you're right. The limit is not on interactions. The limit is on the proteins itself. So 250 proteins can have 3,900. All right. I stand corrected. You guys are right. Oof, you're rubbing it in. Everybody's rubbing it. All right, you're right. I'm not going to argue. Okay. And does your network look like this? Oh man, when I downloaded it, we got 177 nodes and 167 interactions. You guys are having too many. That means you're not doing okay, hold on. Maybe you are wrong and I am right. Have you done the correct point 0.7 confidence also? Did you click on the confidence? Or have you just downloaded everything without confidence also? You have to be very careful that you're doing confidence. Okay, fine. You've done the confidence. All right. So, anyway, this is old information that you see on the screen. So, your confidence is zero point 0.7 or above. If you have confidence zero point 0.7 or above, then you get more genes and more proteins. That's very good. What next? Now, the question is, what about the bone marrow? What about the gallbladder? Let us see. How do you find out the bone marrow and gallbladder information? Of course, it is in the tables. So, let's go to tables. Oh, that's not, the first question was FLT3. Oh, ho. Wait. The question कहाँ गए? Question साथ में निकालने हैं. FLT3 से कितने genes connected हैं? So FLT3 है भी कि नहीं? First of all, search. How will you know which is FLT3 in this huge, fur ball of a network that you have? I told you earlier, you can make a little search by pressing the search window on this little network of yours. Type in FLT3 because that is the question. How many interactors does the FLT3 have in the leukemia network? And you will find that just that region starts to color up for your network. Okay? So here I find that I can count off exactly 6 interactors to FLT3, but this is Purana data. Can you tell me how many interactors you guys found to FLT3? First of all, you have to search FLT3 and then you find the interactors of FLT3. Tell me how many interactors did you all get? Is it 6, just like in my case, which is from the last time I took this class with my students? Is it the true case today also? So you have to search for FLT3 and don't search for it in the original search of the networks. That is the world's search. This network, for searching this network, you have to search in a different search window, this little search window, which is on your network panel. So where your main network window is there is a little search window above that. That will search in your current network. Okay. So don't make the mistake of using the wrong network search panel. So did anybody find FLT3? And if yes, how many interactors did you find for the FLT3 is the question. Let's give it a minute and see if somebody has found something and if you'd like to put it in the chat box. Okay, good job, Preeti. So 6 is 7 ho gaya. From the last time that I did this, I found 6 interactors and you found 7. Is 7 inclusive of FLT3? Or is 7 the number of interactors of FLT3? All right, I'm going to wait for somebody else to give this answer too. All right, maybe you did have 7. I'll agree with you, Preeti, even though none of your classmates have or batchmates have reiterated your result. The search, oh, Krati, the search is right above. You can see it on the screen. Do you see it here? This is your network window. And on top of the network window, there is a little search button. So you just put the name, type the name of whatever you want to search within that. Okay? And when you press enter, your FLT3 will shine up. On the network window itself, you will find the search. That's a good question. A lot of students get confused there. They go back to the original search, which is in the side panel. That is not where you want to go. You've already imported the network. It's already visible on your screen. You just want to search your network, not the whole universe, just your network for FLT3, and therefore you type it above the network right here. And then what do we get next? Next is the question that I was asking, what is the genes in the Palak says after FLT3 nothing changed. What do you mean by nothing changed? I don't understand what has to change. Nothing has to change here. How do we get that number? Oh, by clicking enter. You click enter till it comes and it gets visible, and you press on the node table. So when you get the node table, only the FL33 node will be visible there. So Preeti has done it at 7, and maybe if Preeti is in the room with you, she can tell how to. Kishan Gupta, you found 11 interactors. Very interesting. 11 interactors. That's interesting. Okay. And then tell me something about which gene has the highest expression in leukemia within the gallbladder. So now you have the entire network. This has got nothing to do with FLT3. If you go to the node table, you go to the gene name and press its, uh, um, gallbladder. So go to the gallbladder tissue, and in the gallbladder tissue, there is a little button which does ranking. So it sorts the numbers or the expression values from increasing or in increasing order or decreasing order. So when you put it in decreasing order, the top most expressing gene within the gallbladder will become visible right here. And that gene, as you can see, for leukemia, the gene that expresses highest in the gallbladder is CLTC. I want you to do the same thing for the bone marrow. Instead of the gallbladder, go further down on the node table, find the bone marrow, and for the bone marrow, go and do the expression. And this expression data will not be in your cytoscape unless you had done the enrichment download also. When you downloaded leukemia data, you should have downloaded the enrichment metadata. And this is where the enrichment comes in. So you already know the expression of each one of these genes of leukemia in different organs of the body. And that is why you're now looking at bone marrow and you're expecting कि gallbladder से ज़्यादा bone marrow में expression होगा, but क्या उसी gene का expression उतना ही है, or is it a different gene? So in my case, at least, when this question was built and put together and the answer was searched for the previous time I ran this course, the gene with the highest expression in the gallbladder was CLTC, and the gene with the highest expression in the bone marrow was NPN1. I would like you to answer these two questions. Even if you cannot do it today, do it slowly, do it at home. This is your homework. I'm just telling you how to search for this information here. We find it in the bone marrow and the gallbladder. Okay? And the last question was, which gene has the lowest FDR value in the UniProt database? That also is available in your string enrichment information node table, where you go to the last, because you had downloaded the enrichment data also, go to string enrichment data, and there you search for the UniProt, and within UniProt, you do the FDR value up and down, and you will find that the gene with the lowest FDR value in the UniProt data is the protonco gene. Okay, so that's what you get. So at any cost, this is the solution, but I want you to find the solution. In Wiki Pathways also, you could download the same data from Wiki Pathways and find the answers required. This is the end of day one. I'm so glad that we were able to complete day one. Good. So anyway, I'm going to stick to the same homework question. I've told you the answers, but that doesn't mean that you're going to be, I'll put the question here again. And of course, I think Ruchi had told me that these are, my slides are being screened online. So you can find the question on YouTube, I suppose, as the last slide. So I'm leaving you on the last slide, which is the homework. Please get it done. Tomorrow, please bring some questions. Try it, play around with it, get the answers, get wrong answers, get right answers, doesn't matter. Tomorrow, we will begin with a solution to this step by step. And I hope that tomorrow, when we all come together, you would have those of you who don't have Java today would have Java installed, or at least you would be working with a friend of yours who has Java installed. Does that work for everybody? I'm going to maybe stop sharing the screen now. And come right back. Over to you, Tabassum.
Yeah, thank you so much, ma'am, for this really engaging session, I would say. and the participants. I'm sure that they have learned a lot from this session, and we look forward to learn more tomorrow, ma'am. Thank you so much, ma'am, and see you tomorrow again, ma'am.
Thank you, my dear. Okay. Bye. Bye, everyone.
Goodbye, everyone. Tomorrow we will have session at 3 only for all the participants. It will start at 3. Thank you
Absolutely.
so much, everyone. Thank you, ma'am. Thank you once
Take care.
again.
Bye. Bye.
Bye bye.